She serves for ALAC International as a European Council member and is a member of the Ethical Committee in the Animal Research in Rhineland-Palatinate in Germany. After working as a clinical vet at the German Center in 2004, she joined Covance Laboratory as a clinical veterinarian and animal welfare officer, manager lab animal care, and moved to other Covance sites in the US. So from 2010 to 2012, she worked as an expert in animal science and welfare officer at Merck. And then later on, she served first as the head of comparative medicine and currently promoted as head of animal welfare, facility budget, and technical platforms. Since 2018, Suzanne is an associate director, animal resources and animal welfare officer at AbbVie. Quite an amazing profile there. So Suzanne will ask you to go ahead and have your presentation, a total of 20 minutes, inclusive of Q&A. Thank you.
Thank you very much, Mercy. So, small correction: I don't have a professor title, so I'm just a simple veterinarian. So, it's a pleasure to talk today about zoonotic diseases. So, I'm happy that I have the possibility. Just a short disclaimer: be informed that the views expressed in the presentation are those of the individual Council on Accreditation member based on the experience and not necessarily reflect those of ALAC International or AbbVie Incorporate. So zoonosis, why is it just of high importance? So especially the transmissibility from non-human primates to humans. It is important to recognize that primate species represent significant risk of pathogens transmission to humans in contact, including the collection of samples of laboratory testing and the handling of those samples in the laboratory. So that means we have to follow strict regulations from a biosafety perspective. That either means that we have a minimum of biosafety level 1, so up to, depending on the disease model, maybe up to biosafety 4. So this is from the WOA terrestrial manual. So why are those zoonotic diseases transmissible so more easily from non-human primates to primates? We have a close relationship. So we share a lot of genetic homology. So depending on the species, if it's more New World or Old World or Great Apes, between 75 to up to 98.5%. We have a highly similar tissue structure, similar immune profile, and of course, looking at the physiology or metabolism of the animals and humans, we share a lot of similarities, and especially the host susceptibility to human pathogens. We differentiate zoonosis, so we talk about subroad zoonosis that are those not coming from an animal reservoir, so that's mostly then in soil. So, and then more specific, it's the anthropod zoonosis or zoonosis, depending on if it's the way from humans to the animals or the animals to the human. So as I said, it can be spread by direct contact. For example, by bite wounds, alimentary, so by just bushmeat, aerogenic transmission, or by vectors, so especially arthropods, so ticks, mites that might be transmitting the diseases. So, of course, a problem of being more acceptable or just acceptable to zoonosis. So still hunting of non-human primates and the sale of meat is of great concern, and it is increasing, especially in countries of poorer environment. We see an increasing use of non-human primates in biomedical research, especially with the development of biologicals. So there's a growth of international tourism, ecotourism. So we're all aware about the situation that people go on vacation to see the gorillas in Uganda. So that maybe spreading of diseases also from the humans to the animals. Development of roads, the infrastructure of agriculture, so that just brings us in much closer proximity to the animals. They lose a lot of their habitat, so because of deforestation, on the other side with fragmentation. So that means we are breaching the natural boundaries between non-human primates and animals to humans. So looking at this zoonosis, of course, we distinguish between viral diseases, bacterial, fungal, parasites. Of most concerns are viruses, bacteria, but also a lot of protozoal infection occur, helminths or arthropods. So looking back into the BSE endemic, so that was done due to prion diseases. On the left side, you see just a list of different viruses that are transmitted, and then also listing the vectors. So, for example, yellow fever, measles that can be transmitted by vectors, fecal oral route, for example, hepatitis A virus, which is very common in non-human primates in the field. Body fluids, so rabies by bites, by spreading of just saliva. The B virus, I think everybody is aware of that when you work with macaques, especially with rhesus monkeys. So, and then of course Marburg virus, Ebola, those just creating hemorrhagic diseases. And respiratory enteric, so that, for example, cytomegalovirus, herpes simplex, influenza virus. And then, of course, on the right side, you see more schematic picture of the different spreading of diseases. Classical endemics, so of course rabies, we all know that it's always lethal to humans and to many other species. So I think probably bats is the only one where it's not a lethal disease. Just pest occurred many years ago, so more in the beginning of the last century. But there were also probably 30 years ago outbreaks in India, whether it was transmitted by rodents. Yellow fever occur from time to time. Also tularemie, so which is the Vibrio, and there it's transmitted by mice, and there you see seasonal differences. So maybe there are some years where it's more pathogenic as in other years. And of course, on the right side, you see just how the trade of bushmeat, so in different areas, and how it's then transferred between different geographic regions. Looking back into the 1980s, so when just HIV was just seen for the first time in 1983, it was so the virus was identified and described. So, which you see on the scheme on the right side. So there are different origins, if it's from African green monkeys, or was it from mangabees, so maybe from chimps, but also from drills. And then we distinguish between HIV-1, HIV-2. And the ZIF-MAC, that's an experimental animal model where just the SIV from the sooty mangabees was then just isolated, and typically these models are then operated in macaques, so mostly rhesus macaque, but also cynomolgus or pigtail macaques. So we have other retroviruses, especially the human retrovirus D, STLV, human T lymphocyte virus, fomivirus, which is not yet a kind of big concern. Gibbon ape leukemia is also known being one of the retroviruses. Here you see some pictures. These were in animals that were experimentally infected with SIV, and you see just connected diseases, so that were mostly lymphomas to cytomegaloviruses, so either in the heart, so in the brain, so that causes then tumors behind the eye, or in the nasal cavity. Hepatitis B, of course, we have a good vaccination now on hand. So I think probably like 25 years ago. So it's a hepatnaviruses that is transmitted via blood or sexual transmitted disease. So it causes anorexia, lethargy, jaundice. And there were many cases also described, so either being transferred from apes to humans or the other way around. Monkeypox, I think there was 3 years ago, there were higher incidences. So it is a disease that is known already for many years. It was first described in 1958. So main origin is in Africa, but we have also seen many cases in Europe or North America, especially with trading of monkeys, the use of African green monkeys in Europe when we were just in the stage of developing the polio vaccine in the 16s. So, and this is how on the right side you see this is the disease, how it looked like in humans, that you have this vesicular exanthema. It's not necessarily a lethal disease, probably about 10% of the humans die. So it's more like having a high fever, viremia, just a lymphadenitis, and you see then the erosions, the vesiculars and the skin, but that also occur on the lungs or the mucosal membranes and on the spleen. So there was a higher incidence in North America, I think in probably 15 years ago. That was due to the trade of this Gambian giant rat, where they also think that's a source for the monkeypox virus. When you look at the pictures of the common marmosets, that were cases of cowpox that were transmitted. But of course, that was also then related to a high mortality within the common marmoset. So that, of course, when this is a new world monkey species, so it shows then different susceptibility as probably old world monkeys to the disease. Filovirus, I think we're all aware about Ebola. Just incidences with also is then connected to a high mortality, both in the non-human primates and humans, so which is originated in bats. In Germany in the 1960s, we had an outbreak of Marburg at different test facilities that originate from African green monkeys. And I don't know how much you are aware about Ebola restin. So that's a disease that's lethal just in monkeys, but it doesn't seem to be lethal in humans. And that's what they also used for vaccine trials in the last 10 years. Measles as a morbillivirus, so it's more concern, of course, that animals get infected and it goes along with a high mortality. Routine, we vaccinate the animals before we use them on studies. So if they have an infection, they're immunocompromised for more than 12 months. Typically is this rash that starts at the head and goes down all over the body, so with fever, edema, and perineal swelling. Herpes virus is always a big concern, so especially the B virus or herpesemia, how it is called. So that was first described in 1932. So we have other herpes virus tamarins, that is the hostis, the squirrel monkey, that is just spread into other New World species. You're pretty much aware about herpes papio, just two in the baboon, where it's not clear how pathogenic that is to humans. Herpes simplex, that's mostly then the other way around, spreading the disease to humans. So other cytomegaloviruses, lymphocryptovirus. So we talk about beta or gamma herpes viruses. You see on the top two pictures from rhesus macaques, and this was, of course, I've never seen really in just animals that are infected, any lesions. But of course, in these cases, the animals were B virus positive. They were infected with SIV. And in addition, they got pox virus. And there you see these typically lesions between the mucosal and cutaneous membranes. So this ulceration. And at the bottom, you see the picture of common marmoset. That was the situation that the animals were infected from a human who just had an open simplex infection and was spreading it to the animals. And there it's a little disease in common marmosets. As said, this virus was isolated in 1932, so there was the first described case in China in 21. The origin is in rhesus macaque, so there's no documented case that is originated from, for example, from a cynomolgus macaques. There are a bit more or around 200 cases described worldwide. So I think probably 50 people that died of the disease. That was then just by bite wounds or just saliva, probably urine infected. So I think those who are in the field may remember Elizabeth Griffiths, that was in the beginning of the 20th century, infected and died of B virus. And this is the electronic microscopic picture where you see these typically inclusion bodies that look like a scrambled, not scrambled, so like a fried egg. Other diseases, diarrhea, so rota coronaviruses. So there are also cases of parvovirus. I think that was recently described in cynomolgus, not in cynomolgus macaque, sorry. So, but that can also be easily spread between humans and non-human primates, the fecal-oral route. So if you don't wear proper PPE and just are aware of the biosafety measurements. Other bacterial infections, which are also enterobacterial, is Shigella, which occurs pretty with higher incidences. So that of course can also be spread to humans. Typically, do have these just lesions, just enlarged lymph nodes in the intestines. So, but you also see then like tubercle-like with Yersinia in the spleen and in the liver. So that's Yersinia pseudotuberculosis, which occurs both in Old World and New World monkeys. Other bacterial infection of Klebsiella oxytoca is of concern. Salmonella, so are easily transmitted. Tetanus is more like from the soil, a source of being a disease. Bordetella bronchiseptica, which is more just a respiratory disease. E. coli, Streptococcus, Staphylococcus are typically common bacterial that can be spread from one individual to the other and between species. Tuberculosis, you know that, of course, especially Old World monkeys are highly susceptible to tuberculosis. That's always a concern because it's then even like spread in the colony. Typically, you have the tuberculin skin test and you read it after just 24 the 48 and 96 hours, so that you see that there's a swelling and an edema, and but it seems to be that there's also species difference. So like African greens are probably more sensitive. Cynomolgus monkeys, you have a lot of described cases with multiple negative tests, and all of a sudden you can isolate acid-fast bacterials, and then you at necropsy you see those lungs with which huge tubercle developed over the time. Parasitic infections, so Amoeba, Cryptosporia, they may be associated to other diseases, viral diseases, so they not necessarily cause clinical symptoms, but may be connected to other diseases. They show up. Echinococcus, which is more spread from mice and from foxes. And these are pictures that from my previous job when I worked at the primate center. So that were Macaca silvana that were infected, but also the colony that we housed from of baboons. So that you see these abdominal cysts. They may be connected to the liver, which is the picture on the left. On the right, it was the Macaca silvana. So, and of course, you see within this big cyst, this Golgi cyst swimming around. So even when you open it, it's a high risk that you inhale the scoliosis, and there's almost no treatment. So of course it's more like getting just the prevention, so to keep the animals away from foxes or other animals, dogs that can transmit the diseases to the animals. As said, flagella, Giardia is very common, so Trichomonas ballantidia. They may be opportunistic infections, so they are transmitted by the fecal-oral route, so they cause diarrhea, but also nausea in humans, but also in the animals. Treatment may be very challenging because when you start treatment, so they mimic and hide in the gallbladder, and then they just are shedding active cysts again, so their reinfection is very high. Just to mention some fungal infections, probably Cryptococcus. We see especially with animals that are just feral animals that are caught in the wild. Histoplasmosis, dermatomycosis, porotrichosis, so different diseases. And to say we still have a lot of unknown pathogens that continue to pose a risk to global health. So we now don't know what kind of endemic or pandemic we may see in the future. To mention here are test laboratories. I think right now we have good capabilities. To mention here is virus reference laboratories. So they have different panels for these different species, also looking at African origin monkeys, so African green monkeys or baboons. So when you look at their website, so they list what can be tested for the different diseases, especially when you work in a laboratory setting. So for biosafety reasons to protect your stuff. And last, so there's a revised version of the PHILASA recommendation of health monitoring primate colonies. Of course, it touches mostly those that are mainly used in laboratories, which are macaques, baboons, New World monkeys. So, but it's a good resource and overview also about diseases to be recognized. And that's from my side. So thank you very much for your attention, and I'm happy to answer any questions.
Thank you very much, Suzanne, for a good talk on zoonosis and what we can encounter in primate populations. So are there any questions? We have time for one or two questions. Any questions? Okay, we don't seem to have questions, but I have one question for you. Given that there's a long list of different diseases, are there kits that are optimized to test for multiple pathogens at a time and that are cost-friendly instead of having to test for individual pathogens?
Yeah, of course, it is what I had listed from virus reference laboratories that they have these different panels. But of course, I don't know how the possibilities to have this tested in Africa. So then, because of primates, they fall under the Washington. They are protected. So that means you have to apply always for CITES for shipping samples to the US or other laboratories in Europe or Asia. So that's then always the big difficulties and burdens. So. But of course, I think when you look at the website, you see probably how pricey they are. So, but I think the main problem is the shipping also of the sample. So if you don't have established those kits on site.
Thank you very much. The good thing about this conference is we have those who work in diagnostics, so this is a research gap that we can actually tap into. Thank you. So that's the end of the plenary talk. I'll invite Dr. Claire Njoki to come and lead us to the next session. And for those who don't have seats, there are seats at the front, so please do come in front.
Good afternoon, everyone. I'm Dr. Claire Njoki, and I'm honored to be co-chairing this session with Dr. Mary Kioko. So over the course of the past 2 days, we've had very insightful presentations from the leaders within the various research fields and senior scientists amongst us. I'm honored to be co-chairing this session where we'll be listening to our young investigators, the next generation of scientists. They'll be telling us a bit of what they've been up to. Before we continue, I'd like to request the young investigators to come and sit their seats that have been set for them in front. So perhaps they can make their way. And I've been informed that there's some who have not yet uploaded their talks. So please, could you kindly do that at the back as we continue? Our first presenter. So this session is basically, I think for some of us who are in room 2, we had very good talks in the mid-morning in the area of diagnostics, both for communicable and non-communicable diseases. So this will sort of be an extension of that. And perhaps I could invite our first speaker, Atin, to come and give us their talk. Or perhaps just to clarify, so I'd like to request the speakers to keep their talks to 7 minutes, and then we'll have a session, a question and answer session at the end, which would be about 10 minutes. Thank you.
Okay. Hello, everyone. My name is Adin Diwan. I am a first-year PhD student at Purdue University under Dr. Young Kim, and it's my honor to present here in this conference today. I'm going to talk about non-invasive blood hemoglobin assessment using conjunctiva photography for sickle cell care. So hemoglobin testing. So we do that using initial screening of like RBC production, and it causes like anemia, cancer, hemorrhage, and gastrointestinal bleeding, etc. So currently we use invasive needle-based blood sampling to get an accurate detection, and it may also cause iatrogenic blood loss. But we want to move towards a more non-invasive route. So there are existing non-invasive technologies like a portable analyzer, like ORSense, MBR, EasyCheck, which are basically like take optical signals from fingertips and have some effect on the skin pigmentation, effect of blood flow changes while using them, and require very specialized equipment. There are some mobile health applications as well for this, like HEMA app, AminoCheck, which use a smartphone photo from the eyelid, fingertip, fingernail, and etc. And they do a color-based diagnostics to figure out the hemoglobin. And these are usually very device, light, and format dependent on the image that they're taking. And they require very high color accuracy for this to happen. So I'm going to talk about my overall framework for mHealth, the technology we made for hemoglobin assessment. So we first begin with eyelid imaging, and we focus on the palpebral area, which is the pink area right in your eyelid. And as you can see, we have different diverse acquisition methods. So in any device, using any device, any lighting condition, it is possible to get hemoglobin. So to do that, we want to quantify each of the images into one singular type. So we do that using color correction. And color correction primarily consists of, in today's world, RGB, red, green, and blue. And we want to unify all of that for every image across all devices for all lighting conditions. And color correction helps us with unifying that. Now, once we have those unified images, we have a hyperspectral learning model, which we use to take and learn from them. And after doing that hyperspectral learning, we get the non-invasive hemoglobin computation. And using that, we can detect and get hemoglobin values. So to do this, the primary and the key thing is to have diagnostic color accuracy. So colors can be categorized into three things: color constancy, color consistency, and color accuracy. So color constancy basically means it's identical to us humans from the naked eye, which we don't want for medical purposes. Color consistency is getting the exact same color, but it's not really quite there yet. And for color accuracy, getting the absolute color value, which I'll talk about a little bit. So what diagnostic color accuracy means using diagnostic photography is to detect the exact same colors across various photo acquisition settings. Because photos can be easily be distorted by devices, light conditions, or just file formats. And you can see down below on the bottom left, the light conditions and the file formats. So we can see the sunlight, fluorescent, halogen, how they're very different across different files and across different light conditions. And for the file formats, I have examples of JPEG, RAW, and RAW RC, which we can also see at different time intervals how vastly different the images and the light conditions cause the image to be. And on the bottom right, we can see the device models. So here we took retina imaging of just using a smartphone across different devices, and you can see how vastly different the colors are across each of the same image. So here comes the concept of color correction, or as I like to call it, color recovery. So we do digital image processing technology where we take acquired RGB values that we get from a smartphone image and convert them to CIE XYZ or LAB values. So CIE XYZ is the International Commission of Illumination XYZ values, which are basically numeric values to determine and represent a color. And LAB values are also another color space to determine color values. And these are usually very device, light, and format independent. So as you can see, we have the Macbeth color chart on the top right here. The Macbeth color chart is like a color checker that we use for professional photography. It has a wide range of colors, and it helps to color correct digital photography images. But it's not really accurate because we want to be medically accurate, and it doesn't rectify tissue-specific colors. Generally, it is a very good color checker, but for our purpose, it does not satisfy. So we moved to a more AI method here. We turned towards neural networks for color correction. So we came up with a one-shot learning methodology where we take an image with the color chart we have designed, and using that color chart, we segment the image or the target we want to focus on and the chart. Using a model that we made, we already have the true ground truth values using a spectrometer we took in the lab. And using that values, we can color correct the image by making the model learn the difference between the original ground truth and the photo's color chart values. Using that trained neural network, we apply that neural network on the sample to color correct the whole image, as you can see on the bottom right. And this image recovery method is very unique to neural because it's every photo will have its own neural network trained algorithm and weights. Oh, hello. Sorry. Weights based on the color reference for the best image recovery. Yeah. So color design is very sensitive to our hemoglobin. So we took tissue spectra and we found out the best chromaticity values calculated from a very synthesized spectra. And using that, we narrowed our triangular gamut to get the hemoglobin color-specific values. And using that, we found out the most used colors to get our optimized color chart, which we call hemochrome. And as you can see, using these color charts, we can actually color correct and the images, and all their intensities tend to unify. As you can see on the right image there, all the images with different intensities have equalized intensities after color correction. So here are some original unprocessed photos that we get from the smartphone. And then we apply the color correction process to get these colored recovered photos. For example, in image 12 particularly, you can see how it gets unified to have the most color corrected value across all images. So you must be wondering how I got that spectral data from the eyelid, right? So we use a guiding image to get the exact location of the line scanning. And then we have an hyperspectral image, which gets the average spectrum across the vertical axis, as you can see in the image right below. And using that, we apply that spectral data to our hyperspectral learning model to get the values. So we did our test in Moi University, Eldoret, Kenya for our mHealth algorithm development, where we had 153 total participants, and we used those spectral data and their laboratory blood values to compare our results with them. And then we did a validation study to check our study can be applied anywhere in IU School of Medicine in the USA, where we took 15 sickle cell anemia patients and did their diagnostics to get an inter-measurement reliability. So here are some comparison results between Macbeth color chart and hemochrome, where we see the correlation between just using Macbeth is very significantly low, and when we use a very color-specific hemoglobin color chart, our hemochrome, we get a high correlation and very significant results, and they're within the 95% line of agreement. So in conclusion, hemoglobin-specific color recovery is really important to get accurate diagnostic medical results. And we use hyperspectral learning as a new technology assisted with neural networks and AI to help get a more feasible response. And we also do some spectroscopic hemoglobin computation to get those spectral values. And after doing those validation study on sickle cell anemia patients, we can get inter-measurement reliability and potential for at-home diagnosis as well. And this will help us advance rural healthcare and facilitate effective diagnosis for anemia globally. Here are some acknowledgements from my lab members and Moi University in Eldoret and Gates Foundation for humbly supporting us. Thank you so much.
Thank you very much Atin for such a very exciting piece of work. I'd like to call on our next presenter, that is Hari Priya. Welcome. Please, let's try to keep the presentations to 7 minutes so that we have time for questions thereafter.
Oh, wait. All right. Hi, everyone. I'm Priya. I'm a second-year PhD student in biomedical engineering. I'm from Purdue University, and my advisor is also Dr. Young Kim. And my presentation today is going to be on smartphone imaging of conjunctival microvascular changes. And we want to see if we can actually quantify these changes and if they're associated with high blood pressure in Kenyan pregnant women. In Kenya, 20% of maternal deaths are caused by hypertensive disorders during pregnancy. So in this map here, you can see the major urban centers that are located in Kenya. And actually, these are the areas where antenatal care is most accessible. And areas outside of that is where the maternal mortality is the worst. And so these hypertensive disorders, it affects rural women the most. But what if a pregnant woman could just check if their blood pressure is high just by taking a smartphone photo of their eye? And the way that we can accomplish this is actually looking at the white of the eye. And the vessels are plainly visible there, a lot of the microvascular vessels. And so we can segment these vessels and then study microvascular metrics like vessel diameter index as well as tortuosity, which have been shown in previous literature to be associated with high blood pressure. So for this study, we recruited pregnant women from all trimesters from Moi Teaching and Referral Hospital at Eldoret. And we had a total of 405 participants, a subset of which had high blood pressure. And we captured smartphone conjunctival photos with 3 different smartphones. And we also collected some basic demographic as well as clinical information, including about their pregnancy, as well as their general health and medical history, and blood pressure, as well as BMI. Next, we processed these photos using an algorithm to perform a conjunctival vessel segmentation, as well as to get a skeleton of the vascular tree using the center line. So from analyzing these results, we found that there was a significantly reduced vessel diameter index, as well as an increased tortuosity that was observed when comparing the normal blood pressure participants with the high blood pressure participants. And it's crazy because we're just doing this with a smartphone photo, and we're able to quantify these differences very significantly. Here are some representative images. Overall, the high blood pressure participants had observable venular constriction as well as increased curvature of their vessels, which has been shown in previous literature as well, but not with smartphones. And I also wanted to point out these brown deposits that a lot of high blood pressure patients had in their eyes. And this is also a typical feature that's present in high blood pressure, where it can cause leakage of blood from microvessels. Next, we grouped these patients based on their weeks of gestation, so looking at patients who were less than 20 weeks and past 20 weeks. And actually, when we look at the vessel changes before 20 weeks, there's barely any changes. But when we look past 20 weeks, there is a significant difference in both the diameter index as well as the tortuosity. When we look at just pregnant women with normal blood pressure, we actually can't see any significant vessel changes that are associated with the weeks of gestation. And so what that tells us is these particular changes that we're observing is very specific to high blood pressure during pregnancy, and not just the cardiovascular challenge that pregnancy can put on a woman's vascular tree, so which could also cause changes, but we can't see them here. Next, we also tried grouping these participants by age, and actually we saw the most significant curvature vessel changes in the youngest patients as well as the oldest. And this also is in line with what we know of hypertensive disorders and risk of hypertensive disorders in the youngest and oldest women. Finally, we can use these vessel changes and we can actually train a logistic regression model to be able to predict which of these patients have a blood pressure elevation. Just using the vessel features, we were able to achieve around a 0.74 AUC. And if we include BMI, which is a significant predictor of high blood pressure, this increases a bit more to 0.78. So overall, in conclusion, this is the first smartphone photo-based study that directly quantifies blood pressure-associated microvascular changes just by plainly looking at the conjunctiva. And these vessels are significantly altered from high blood pressure during pregnancy. And a lot of the vessel changes that we see in regards to the weeks of gestation as well as age agrees very well with known preeclampsia and gestational hypertension risk factors. Overall, there was also no differences in the results when we compared the 3 smartphone models that we captured photos with. And these vessel changes can potentially be used to screen for hypertensive disorders during pregnancy from just a smartphone photo of the eye. And here are my acknowledgements. We have an amazing clinical team at MOI, and I also want to acknowledge my lab and funding sources and references. That's all. Thank you.
Thank you so much for that wonderful presentation. So I'm going to call upon our next presenter, Mr. Harrison Waweru.
All right, thank you so much for this opportunity. My name is Harrison Waweru. I work at Mount Kenya University in a lab called Gitaka Lab, where there is a center for research in infectious diseases. Our work majorly, we focus on maternal health in terms of diseases and ailments that affect the health of the maternal, the health of the mothers and the health of the babies. So from our earlier presentations, you may, for those who are in the morning session, most of the presenters from our group presented regarding placental malaria. So in this presentation, I'm going to show you some work that we are doing in developing a point of care detection system that is a paper-based assay. So in this presentation, I want to talk about the burden of sexually transmitted infections. These include infections that are caused by bacteria, such as Neisseria gonorrhoeae, Chlamydia trachomatis, and vaginalis. These diseases affect the maternal health of most women negatively. That might lead to stillbirths and might also affect the babies before they are born. As shown in this map, the highest burden falls in the sub-Saharan Africa, where our country is currently located. So from this burden, most women do not have access to these diagnosis systems, mostly in those poor resource settings where we don't have the necessary equipment, and also we don't have the personnel that are required. And most of these women get tested when the diseases or the infection has spread so far, instead of it being detected early. On top of that, not to mention that we also have a challenge of antimicrobial resistance from these bacteria. So most women just get prescribed medicine from their chemist, and most of these medicine may not work. So from our study, the background of our study, we were currently trying to solve the issue of access to diagnostics in those peripheral health facilities, and also to introduce the concept of freeze-dried, that is lyophilized assays that can be used in the resource-limited settings. So our aim here was to develop an isothermal nucleic acid analysis technique that can be used to diagnose these infections across from Neisseria all the way to Trichomonas vaginalis. So this slide mainly I included for those students or members of this meeting that are not familiar with LAMP. Just to show you, the concept of LAMP is that it utilizes more than 2 primers, normally which we do in normal PCRs. And these primers enable for rapid amplification of nucleic acids. So for the detection or interpretation of the results, usually on a gel electrophoresis, you get a ladder-like pattern instead of bands. This shows positive reactions. This is due to the amplification of different primers that produce different lengths of DNA. Other detection methods that can be used to confirm results include turbidity and fluorescence. And in this case, we are going to concentrate more on colorimetric or color change that includes incorporation of dyes. So LAMP in a nutshell, it has been developed since a decade ago. And this slide is usually to show the many applications of LAMP that are out there that have been utilized. So we are not trying to reinvent the wheel. So many other studies have tried to develop this paper-based amplification. And what we are trying to do is just improve on this detection and then incorporate this into our own setting, that is the resource-limited settings. So the concept is simple. The first step, which is the DNA extraction, majorly this one we have been able to accomplish in our lab, where we can use some reagents that are used just to lyse the bacteria, and then you have your nucleic acid suspended in solution. Then the most important part is to have our LAMP amplification reagents incorporated with a dye. In this case, we are using the hydroxyl naphthol blue dye, or in short, we call it the H&B dye. The idea is to lyophilize these reagents on a piece of paper. In this case, we are using electrospan fiber mats, and then perform LAMP amplification on this paper alone, and then have a detection that can be observed by eye. Or if we move further with this study, we can also incorporate a lateral flow. The idea is then to incorporate all these reactions into a prototype, which we'll build as a microfluidic device that can be used to perform this detection. So you might ask, why use electron span fiber mats instead of just normal papers? So the special thing about fiber mats is that they have nanofiber size of threads, and they have a high surface area. So one important thing to notice is the porous structure of these fiber mats. They allow the reactions to happen within their nanofibers. So the first thing we did is take scanning microscopes, images of these fiber mats. So we labeled them as 8951 and 6613. As you can see, they have different length of, different wings of the fiber mats. This is just another comparison, just to show how different the fiber mats are from the cellulose Whatmine papers that we are used to. So we went ahead and did a session of the LAMP reaction using the Neisseria gonorrhoeae DNA. And as you can see there, the performance of the fiber mat 8951 was more than the tube-based assay. This was just to assess how the different paper matrices will perform. Then we went ahead and performed a colorimetric tube-based assay just to see if the dye will work. And as you can see, due to the depletion of magnesium ions, this leads to the H&B dye to change from violet to sky blue, which confirms a positive reaction. Then we went ahead and incorporated this colorimetric assay on the fiber mats themselves. The color changes you see there are just paper dots. So we punched these fiber mats into paper dots, then we incorporated the reagents on the papers, and just to see how they react. As you can see, they supported the reaction. So we went ahead and performed a specificity assay, and as you can see, our primers are specific, and the assay showed no cross-reactivity with the other bacteria. Then we did a sensitivity assay, and we were able to detect as low as 10 copies of DNA per microliter. So in a summary, these electrospan fiber mats, there's an error there. Electrospan fiber mats successfully supported this assay, and distinct color change was visible at 10 copies per microliter. So we conclude that these fiber mats are suitable for field use, and long-term reagent stability studies are being conducted in our lab. So in conclusion, our findings suggest that these fiber mats can offer a portable and reliable lamp-based diagnostics. So the ongoing work for now in our lab is to develop this paper-based POC prototype and also test the shelf life of these lyophilized reagents in the field. So I would want to acknowledge Science for Africa for their funding, and my lab, Gitaka Lab, and Mount Kenya University. Thank you.
Thank you so much, Harrison, for that wonderful presentation. I would like to call upon our next presenter, Mr. Douglas Gifu. Let's applaud him as he comes on.
Maybe a small announcement. We're going to be grading our young investigators based on their presentations, and then also based on the responses to the questions from the audience. Also, a reminder that keeping time is one of the parameters that will be used to score your general performance. So let's try and observe that, please. Thank you.
Okay, thank you. That starts with me. So, I can do the first slide. I'm Douglas Nganga from Gifu University. Yeah, and I'm going to present on spiral deepening of microtubules actively driven by kinesin motors. And those who were in the room B, we had some presentation on spiraling, active spiraling of microtubules. So here I show the kinesin motors that are in the cytoskeletons, and you can see them carrying vehicle and transporting materials on microtubules. And they do this by hydrolysis of ATP, where they are able to convert that chemical to mechanical, to motion. And so, in the next slide, I show the inverted motility assay, where now the kinesins are the ones that are driving the microtubules, and they just move randomly, and they demonstrate the active polymer dynamics. But these can be applied in molecular shuttles, where you now use the microtubules to transport cargo, because a little spacious and something like that. So based on this, we see that microtubules are important, and it's important to know the mechanics of microtubules as active polymers. And several studies have been done to measure the mechanics of microtubules using passive methods, where we have thermal fluctuations, and active methods, where we have manipulations like with active tweezers, optical tweezers, or magnetic tweezers. But this hasn't done any better, as you can see the list of results that have been obtained range all the way from around a piconewton per piconewton micrometer squared all the way to 22 piconewton micrometer squared. So that's still a wide field where everybody wants to try and measure the mechanics of microtubule. And we see that in this active polymer dynamics, we see that if we have some boundary conditions like clamping or pinning, we see flagella like beating, and in the same case, we can see buckling and spiraling. And these dynamics are governed by the interplay between the filament bending rigidity as well as the motor mechanics. So my objectives is to reveal the mechanism of the spiral deepening, where the spiraling occurs and then it's released, and also to demonstrate how this can be used to infer the mechanics of the microtubule through computer simulations, where we have advantages of computer simulations, like you can choose where to do the pinning. So our previous study, we did some, we showed some phase diagram of the motor surface motor density and the length of the filament, and we showed a boundary like was presented in the morning. And I used this to get some parameter choice. So in this case, in this study, I'm using 20 micrometer and 20 motors per micrometer motor density. And in this case, we see now the movement of the microtubule, which is the brown, the orange. And the green motors are the ones that are binding, and you can see it got pinned and it depinned because we added the depinning threshold. And the simulation method is as shown, like bead rod polymer with spring-like motors. And you can see where the depinning occurred and the filament is almost straight. But we have another case where we lowered the bending rigidity of the filament, and you can see that it formed a spiral, and after spiraling and rotating around, remember I said that these green motors are the ones that are binding, and it's able to get released. The spiral depinning occurs similar to the one that I showed about by Phillips et al., and you can see that it gets released and it continues in another trajectory. And definitely in the application in biophysics, then that's interesting to study what occurs. And to study why this is the case, we do evaluation of the pinning force, the force that is preventing the translation of the filament in the time evolution. And in the right, I have the conformations at the different points I have shown in the pinning force profile. And you can see we have one prominent peak and then steady state fluctuations. But when we lower the motor density, the bending rigidity, we have the initial peak, which have reduced drastically, and then we have steady state fluctuations where we have peaks that are greater than the initial peak. So in this case, we have a model to study the initial peak, and in this model, which was presented in the morning, we have, this is during the occurrence of buckling, and if we measure the F peak, which is the peak at the tip, then we have this equation here of the F peak, and we use that to plot together with the other initial peaks, and we see that they agree, as well as when we plot with the other time average of the pinning force during spiraling, we see that it's less dependent to the bending rigidity in comparison to the initial peak. So we study why this is occurring in high bending rigidity and not in low bending rigidity. And we realize that in low bending rigidity, the red curve there shows the direction of flotation. So in low bending rigidity, due to the deformability of the microtubule, then it's able to change the direction. And during that time when it's changing the direction, we have the force lays, and we checked this with the number of motors during near the pinned head, and we realized that there is a big correlation between the forces and the number of motors binding near the pinned head, but less correlation with the motors binding along the whole filament. And you can see this is just to show that the depinning force is selected, and in this case, it will always depin before spiraling have occurred. But for the low bending rigidity, then it's able to depin during spiraling, and we can reproduce, and this was the conformation during depinning. We can reproduce what was seen before. And we can use this approach to infer the bending rigidity with high throughput, because we are able to access this kind of behavior using normal microscopes. And you see for the application in molecular shuttles, where you can see the molecular shuttles being used for the testing and detecting of analytes. Thank you. Yes.
Thank you so much, Douglas, for that wonderful presentation. Allow me to call upon Patrick Wakaba to give us his presentation. Let's applaud him, please.
Hello, everyone. My name is Wakaba Patrick Minor. I'm a second-year master's student at Nagasaki University in Japan. And today I'm going to present on some of the work that I've been conducting as part of my master's research work. So the title is a palm-free CRISPR-Cas12-based method for the detection of Mycobacterial tuberculosis. So in recent years, there has been an increase in the development of CRISPR-Cas-based diagnostic assays. So you find that in the conventional CRISPR-Cas12-based detection approach, so what happens is first you identify your target organism, then you get the genome. Then within the genome, you identify a target gene. So mostly this gene is the conserved gene within the organism. Then from the gene, you identify what we call a protospacer adjacent motif. This is a PAM sequence. So this is a sequence made up of diamine, and any other nucleotide. So then adjacent to the PAM sequence, you identify about 17 to 24 base pairs and use that to design what we call a CRISPR RNA. So the next step is signal amplification, where you use target-specific primers to amplify this region. So with the amplification, you produce many, many products that can be used to activate the CRISPR enzyme in presence of the CRISPR RNA. So after activation, the collateral cleavage is also activated, and you can cleave any single-stranded DNA molecules, including the reporters. And this can then be visualized either by fluorescence or lateral flow strip. So you notice that in this conventional approach, if the sequence doesn't have the protospacer adjacent motif sequence, then it becomes difficult to apply a CRISPR-Cas enzyme diagnostic method. So another gap in this conventional method, yes, you have the PAM sequence, but at times you design a CRISPR RNA, but this CRISPR RNA is less sensitive or it's less efficient. So this means in some guide RNA, yes, it's working, but the signals you get are very low, and this might reduce the sensitivity of the assay. So we saw a gap in this, and we developed some assays that can help solve this challenge. So we developed 4 different assays. These assays are aimed at producing a CRISPR RNA. So the first assay, we call it the two-tailed primer assay. So in this assay, we tailed our target-specific primers with a T7 promoter sequence, and our reverse primer, we tailed that with a CRISPR RNA DNA sequence. So upon amplification, you can get the T7 promoter and CRISPR RNA DNA sequence in the products. Then from that, you can do transcription using a T7 RNA polymerase and get the CRISPR RNA. Then from there, you can activate the Cas enzyme and detect that using real-time fluorescence or endpoint fluorescence. So the other assay is called a 3-tailed primer assay. In this assay, we use 3 different primers. So the first 2 target-specific primer, we tail them with a T7 promoter complementary sequence. Then after amplification, you introduce a T7 promoter sequence in the products. Then in the subsequent application, you can use a primer we call the CRISPR RNA primer. So this is made up of a T7 complementary sequence and a CRISPR RNA DNA sequence in red. So this means that you can produce the T7 promoter sequence and the CRISPR RNA in the products, and then you can transcribe that to produce the CRISPR RNA on both strands. So the other 2 assays are derived from the 3-tailed primer assay. So in this, you can either tail the You can either tail the reverse primer and leave the forward primer untailed, then use that primer, which is the CRISPR RNA primer. Then you can produce the CRISPR RNA from one strand of the products, and that can activate the Cas enzyme, and then you can detect the cleavage, either real-time or endpoint. So this also is the untailed reverse primer assay, where you can tail now the forward primer and leave the reverse primer untailed. Then you can use that primer, the CRISPR RNA primer, and then you can get products containing the T7 promoter sequence and the CRISPR RNA, which can then be transcribed to produce CRISPR RNA. So this, with all the 4 assays, you only get the CRISPR RNA when the amplification takes place. So without the amplification, you can produce the CRISPR RNA. And also within the target sequence, you notice that there is no PAM sequence. So we started by testing the target-specific primers. So we used the target-specific primers without the tails and amplified Mycobacterium tuberculosis IS1081. So these we used genomic DNA from the strain H37RA. So you can see that with the target primers, the band is lower. But if you use the tailed primers, the bands will increase because you're increasing the number of nucleotides. So we performed PCR for about 1 hour, 15 minutes. Then after that, we transferred the 2 microliters of the product to 1-pot transcription and CRISPR. So we monitored this. So we incubated this at that 7 degrees and monitored fluorescence real time. So you can see, actually even within 5 minutes, you can see signal. And that, those are images taken, fluorescence images taken after that means period of incubation. And you can see that it's only when you have the tail primers that you can see the fluorescence. The negative, there is no fluorescence. So next, we did the limit of detection. So this is for the PCR, transcription, and CRISPR-Cas assay. So we did the limit of detection. So this is after optimization of many components. So we did limit of detection, and from this, we could detect up to 200 attomora in samples. So because of the time and resources, we decided to work with just one assay from the 4 assays. So we used this assay to try and develop a point-of-care application. So in this point-of-care application, we decided to change the amplification method from PCR to NAMP assay. So we took the 4-adenine primer with that 7 complementary sequence, and also use the CRISPR RNA primer. So with this, you can get the CRISPR RNA, and the advantage is that you can use a heating block to do the incubation. So we performed limit of detection, and we can also see from the NAMP-based assay, we could detect up to attomolar concentrations. So next, we did specificity test. So in this, we, apart from doing in silico specificity testing, we tried using the primers and tried using the primer to amplify non-tuberculous mycobacteria, and also some other bacterial strains, such as Staphylococcus aureus, E. coli, and also human DNA. Because you're using human sample, you might have the human DNA in the sample. And you can see it's only when you have the mycobacterial tuberculosis that you see the signal. So in conclusion is that here we develop 4 palm-free assays, and these assays have a point-of-care application. And from the discussion, you can see that there is high sensitivity up to attomolar concentration and also the specificity. So this work is still ongoing. So I'm trying to develop a 1-pot NAMP-assisted palm-free CRISPR assay. And also I want to continue to clinical validation, where I want to compare the PCR-based assay, NAMP-based assay, and also compare that to qPCR. So finally, I'd like to acknowledge my team members, so including my supervisor, Professor Nobuo Saito, my assistant supervisor, Professor Naoki Uno, and also Nagasaki University faculty, Mext and Shonogi, who are the founders of this project. And finally, I'd like to thank the AIBBC for giving us opportunity to present this work. Thank you.
Thank you so much, Patrick. And on to our last presenter of this session. Please help me welcome Dr. Rachel Kimani.
Thank you very much. As we begin, all of us here have received an antibiotic prescription at least once in our lives. And therefore, this is very relevant, and I hope to keep you captivated for the next 7 minutes. So we did some work on resistant pathogenic bacteria from clinical and hospital and samples in a referral hospital in Kenya. So unless action is taken, rising drug resistance and burden of AMR will balloon to 10 million deaths each year by 2050. Cumulative cost to global economic output will reach 100 trillion USD. From the world's most comprehensive review on AMR, it was published in The Lancet. we find out that AMR is still a global crisis. It threatens modern human medicine, affecting all regions, especially in African setting. From the review, it was found out that in 2019, 4.95 million deaths were due to the fact that the resistant infections played a role, even if they were not the sole cause of death. Also, 1.27 million deaths were directly attributable to bacterial AMR. That was in 2019. Therefore, there is need for surveillance, antimicrobial stewardship, infection prevention, in order to avert this problem. Currently, we have a rapid rise in stagnation in new antibiotic classes discovery. From the slide, what we see is that there's a decline in the discovery of new classes over time. Previously, we had a golden era between 1940 to 1970, whereby many classes were discovered then. But currently, where we are, especially after the 2020s, no new classes are being discovered. And this can be explained by many factors. For example, funding now is mainly to the anti-cancer drug development. Therefore, scientists are no longer motivated to look for new antibiotics. Also, most of the projects in early development phases, those which are concerned with discovery of new antibiotics, end up in the valley of death. We were able to see that here in the morning. The valley of death, whereby the product does not go beyond the early phases due to lack of funding and support. So we did some work at Thika Level 5 Hospital with our group, and we collected clinical samples and surface swabs. We did the routine microbiological isolation of the pathogens and also did antimicrobial susceptibility testing. Identification of the species was through VITEC, and later on we were able to do genotypic tests whereby we looked for resistant markers using PCR and also proceeded to do whole genome sequencing for a few selected multi-drug isolates. So for some of our findings, we were able to isolate E. coli, Klebsiella, Acinetobacter, Enterobacter species from these samples. And one of our findings was that the proportion of resistance varied across the different hospital units. There was a declining trend in proportion of resistant isolates from the intensive care unit to high-touch surfaces. Notably, Acinetobacter baumannii exhibited the highest resistant rates in all units. The mean age of our participants was 46 years. And for the percentage resistance across the different antibiotics, imipinem and meropenem, which are the last resort drugs, demonstrated relatively significant resistant levels. This shows that emerging resistance is being recorded here in our country. As we did further analysis, we were able to see that A. baumannii exhibited the highest resistant levels, ranging from 80% to 90%, and therefore this is a pathogen that we should look out for. Sorry. This image also depicts that A. baumannii isolates emerge as the most resistant pathogens. We were also able to demonstrate gene cassettes in the integrons after whole genome sequencing of our multi-drug isolates. Notably, we were also able to show plasmid replicons. Therefore, the resistance was carried in plasmids for most of these isolates. So as I conclude, the high or elevated resistance of A. baumannii against all antibiotics is concerning and may indicate health-acquired infections. For the carbapenems, which are the last resort drugs, they still show some effectiveness, but resistance is emerging. These findings highlight the need for targeted antibiotic stewardship and routine surveillance in our community. Thank you.
Thank you so much, Dr. Rachel, for that wonderful presentation. Ladies and gentlemen, please let's give all our presenters a round of applause. Yeah, so we're going to move into the next session where we're going to have the question and answer session for all our presenters. Kindly let's be engaging and be as tough as possible. Thank you so much.
Okay, any questions from the audience?
So, my name is Samuel. My question goes to Patrick Wakaba. And thank you for the nice study that you're conducting. And it's insightful and a breakthrough that you are able to use palm-free to conduct a CRISPR-Cas detection of tuberculosis. My question is, do you foresee a situation where we could be able to replicate that and use it in other detection of other pathogens? And is it possible also to multiplex and use just one sample and several CRISPR-Cas that are palm-free in one reaction? Thank you.
Okay. Thank you for your question. So in this work that we are doing, it's possible to replicate what you are doing in any other organism. So all you need to have is like the PCR, the normal PCR primers, and just tell them with artisan promoter sequence and CRISPR-RNA DNA sequence of your choice. So if you know like this CRISPR-RNA and double-strand DNA have high affinity and they give good results, you can just use that already CRISPR-RNA already known, then use the same sequences, tell them with to the target-specific primer, like in the PCR, and then you can detect any other organism. So in terms of multiplexing, I think it's still possible. I think it depends on how you plan your assay and how you go on developing your assay, but it's possible.
Anyone else?
Okay, thank you so much. My name is Anthony. My question goes to, I believe, Speaker 3, that is Mr. Harrison, about your paper-based lump assay. I've seen that it has a great potential in point-of-care diagnostic. My question is, what would you say is your biggest challenge in translating this innovation from lab to a reality clinical and community setting, especially in low resource settings in Kenya? Thank you.
Let me get, to get your question right, you are asking about the challenges in deploying this in the field?
Yes, I'm asking if, what is your biggest challenge in deploying this very good innovative idea into the resource setting areas, especially in low resource settings in Kenya or Africa in general?
Well, thank you for your question. As for any assay that you want to include in the health system, some challenges, most of the challenges are usually technical and sometimes implementation. For technical challenges, remember we want to develop a device that can be used in settings that don't have resources like we have here. So one of the major concerns that is technical is the reagent stability in the field, and also in modifying the stereotype, that is miniaturizing the assay into a small detection thing. The other thing that can be a challenge is the users themselves. As for now, we are using colorimetric. So color interpretation varies from one person to another. So we have to come up with a standard way of getting these colors right. For men, most of the men, I think they are colorblind. So interpreting colors becomes a challenge. So we might have to provide some color charts. Also, we might also have an app that is incorporated with AI that can help you interpret these colors well. As for implementation challenges, of course, we have some regulatory bodies. You just can't integrate an assay into the health system without their permission. And other challenges, you meet them along the way and you get the solutions on the ground. Thank you.
Okay, thank you, Harrison.
Okay, thank you. My name is Paul Kosio from Maseno University. My question goes to the presenter who did something on an invasive method of hemoglobin estimation. I wanted to understand which fraction of hemoglobin were you measuring, or you measured all the fractions, the total hemoglobin. Then again, did you compare it with the gold standard? Because we know that there are several methods of HB estimation, of which cyan methemoglobin remains the gold standard as recommended by the International Committee on Standardization of Hematology. Secondly, or thirdly, if you compare, considering the physiological status, if you look at the eyelid, we know there are factors that may affect that. Maybe someone has cried, and after crying, do you still want to evaluate HB in the same way? Thank you.
Thank you for your question. Yes. So for doing comparison, yes, we do have compared to the clinical values that we get from blood work. So we did. So we had Moy University help us get clinical laboratory values and get like the total hemoglobin value, and we compared it with our total hemoglobin values. So what we basically did was we took images from both right and the left eye, and calculated using our model to get our predicted hemoglobin values. And by averaging those two out, we get our total hemoglobin values, which we say is within plus or minus 2 grams per deciliter as determined by the factor of WHO. And yes, we do compare it with the clinical values. As for the second question, for the crying part, we did a study with pediatric infants where we did have a lot of babies crying. So unfortunately, we did not use those photos because that might lead to hindrance and any sort of like any eye disease or presence of eyelash within the photo, we had to remove to get the best optimal picture of the eyelid and use that as our reference for that calculation for hemoglobin. I hope that answers your questions.
Thank you, Atin.
Yes, thank you. This is Mark Macau, and my question goes to the last presenter, Dr. Rachel. I believe that with there's so much, like the raise in AMR, the AMR raise is really high. What's your take on the use of bacteriophages as like the new method to curb antimicrobial resistance? Thank you.
Thank you. Thank you, Mark, for that question. About the use of bacteriophages to avert the problem of AMR, it's still one of the ways we can explore in order to overcome the declining antibiotics that are no longer, whereby the bacteria are no longer responding to. So concentration from sewage and subsequent downstream experiments to see if they're able to lyse these hypervirulent organisms, then still stands as one of the areas we continue to explore.
Thank you so much, Rachel, for that. Do we have any other questions?
My name is Makena Moniki from Mount Kenya University. My question goes to Haripriya. Given that hypertension is it's a challenge to pregnant women, how feasible is it to incorporate the mobile-based monitoring approach in the antenatal care or in the community?
Thank you for your question. There are some different considerations that we need to have. Actually, as I was preparing for this presentation, I was looking at the prevalence of smartphone use in various regions in Kenya. And in certain regions, they are still low. And so we know that smartphones are common and they can be present in clinics. And so some coordinators, clinical coordinators, or perhaps someone like a community health promoter might have a smartphone, and so when they go into the field, they may be able to use that for screening. So we can think about that from a translational standpoint. I think from a technical standpoint, there are still some challenges to navigate for a technology like this, because there are a lot of confounds that could be present, because we know that there are many diseases or many factors that could affect vessels. And especially we're looking at a very obvious surface as well. And so I think that is another specific technical challenge. But I think surely a tool like this would improve accessibility even in areas where they may not have internet. And so I think it can help with identifying some cases of maybe hypertensive disorders that are emerging early on before a mother might go in for her first antenatal appointment, which may be a bit later in the pregnancy. So, thank you.
Okay, thank you, Haripriya. I'll allow space for just 2 final questions, please.
Okay, my name is David Mora. My question goes to Hamad. So you mentioned that you're using smartphone, and we understand that different smartphones have different megapixels, and that might affect the quality of the photo. So did you factor in that into your analysis? And then my second question, maybe I might have missed. You have talked about neural networks, but you have not given the examples of the neural networks that you have used. Thank you.
Hello, thank you so much for your question. So yes, we took a variety of smartphone models. So we used the models like Samsung S21, A52, and Google Pixel 6. So we wanted to take a high-end model, S21, a low-end Samsung model, A52, and a more common model, which is Google Pixel 6, to see if we can get a different variety of images across different devices. So during my talk, when we talked about being device independent, that's where we use different devices to find out what differences there were. But doing color correction on those images tends to unify all of those color values that we want to look at to get those accurate hemoglobin values. So we do, yes, we did comparison, and they were mostly relatively equal to all of them, because color correction tends to color correct all of the values, leading to a more diagnostic value. As for your second question about neural networks, so we made our own neural network. We did not pre-made neural network. We trained a very small network that we in future want to be able to run solely on a smartphone. So in case, because in resource limited settings, you're not going to be able to get a high-end GPU to run millions of GPU hours. So we are trying to compact our neural network into a model that is just able to run on an edge device, and that is an ongoing study for now. But we did create our own using Python, and it is a very well and working model. Thank you for your question.
Thank you, Artin.
Good afternoon. Now, I have two questions. The first one goes to Haripriya. Now, about testing for BP, you've used it on pregnant women. Just a thought, can we scale it to all the other people? Because in our societies we have a lot of people who are affected with high blood pressure. Is it possible that we can downscale it and use it in our normal day-to-day activities? That is number one. My second question goes to Dr. Rachel. Now, AMR, we've said it is a key thing, and by 2050 it will be more chaotic. What are some of the surveillance measures that we can have to be able to reduce the effect of AMR in our society? Thank you.
Hi, that's a great question, and that is a thought that I had as I was working on this project. It is possible, for sure, because we know that actually there's a lot of previous literature that supports the idea that we can see similar changes with just general hypertension. Actually, there's very few studies that have been done specifically in pregnant women. I think this is one of the very few that have been done looking at microvascular changes specifically in pregnant women. And so I think it is definitely possible. We would just need to find the right cohort to be able to test that out.
Okay. Oh. Last question now, as we close this session. I think Dr. Kimani would answer the final question, and then we can proceed to the next session. We still have time to have these discussions even during breaks, and as we're still here tomorrow. So I think because we need to proceed, we can just close with your final answer, yes.
There was a question for me, maybe I can answer that.
Sure.
Yeah, so what we can do to avert AMR is continuous surveillance, because in the first place, we have to know the burden before we act. For example, in Kenya, the picture is fragmented because surveillance is not optimal. And when you talk of surveillance, this goes up to molecular level. Secondly, there are other measures such as infection prevention control, antimicrobial stewardship, which are now being implemented, taking shape in our country in an effort to combat AMR. Thank you.
I realize there's one young investigator who we haven't heard from, Douglas. So there's a final question for Douglas.
Yeah, for me, I have a question to Haripriya. Three questions.
Sorry, does anyone have a question for? Please, let's continue this after, during tea break. Any question for Douglas? From the audience, please.
Thank you so much. I mean, my question is very general. What challenges did you experience when you were doing your work?
Okay, thank you for your question. Actually, that's what happens when you have biophysics sandwiched between diagnostics, and I was expecting something close to that, but thank you for your question, and I will meet you for your question. So, our study is mostly, we are doing some computational study on how to extract physics from biology. So we are, in the cell, there's a lot of activities that happens, and then we harness that for fundamental study, and then later, that is applicable in engineering applications, like, for example, I showed something to do with the molecular shuttle, and that molecular shuttle can be applied to deliver something. For example, you can use it for in diagnostic stage for detection of antibodies and something like that. So one of the challenges is mostly because in the motility assay experiment, I showed a lot of random movement of the microtubules. And so in the experiments, it's difficult to perform the boundary conditions like pinning, and so that you can observe that. They just happen randomly. It can be pinned at the body or at the end. But now for computational study, it's a little easy because you detect where exactly you want to pin, and when you pin that, you get the very good analysis. And once you find out the physical conditions that you want to perform your now experiment, you can easily predict. So what happens in the experiment in near future after you have the computational study, it's already having a path through predictions performed by the computational study. So we have challenges yet because our computational study is based mostly on what have been extracted from experiments. So you need experiments to have more results and to be able to get the parameters that we use for computational study and to imitate more of what is happening in the cell. So we need more experiment, but the experiments need more computation. So it's potato. Thank you.
Okay, let's give our young investigators a big round of applause, please. So I'd like to thank you also as the audience for your participation in this session. I'd like to thank my co-chair, Dr. Mary also, and I'd like to invite Dr. Ruth to lead us through into the next session. Thank you.
I think that was a very exciting session. And then I think we're still going to listen and learn more from the young investigators through the poster sessions, which they've already been set up. So please engage with the young investigators, go see their posters, go talk to them, and we will be able to learn. We are running behind schedule, so I'd ask that we, even as we do the posters, we can as well, at the same time, take the health break. But let us be back by 5 p.m. And as we all go, I'm kindly asking the following persons to remain behind briefly. Professor Yan Hao, Dr. Sarah Kitui, Professor Stephen Shapiro, Dr. Paul Mireji, Dr. Evans Egwin, Dr. Stella Irungu, Dr. Claire Njoki, Dr. Paul Mireji, Dr. Peter Fundi, Dr. Isaac Ngere, Professor Wahungu, Dr. Fahad Zamani, Dr. Joram, Dr. Bernard Kanoi, Professor Yan, Dr. Clement Shiluli, and Dr. Dishon Muloi. Thank you very much, and we can take a break.
Good afternoon, good evening. I would like to call the meeting to order so that we start our evening session. Let's settle in quick, please. So welcome back. I'm chairing this session with my co-chair, Dr. Sarah Kitui, and it's about NTDs. NTDs is a group of infectious diseases that primarily found in low-income populations, and they're called neglected tropical diseases, maybe because they lack funding or the people are neglected. But we should also ask ourselves why maybe this afternoon session, late in the afternoon, we are this time slot, because they look at the NTD session as something that is not so important. But I do think that's not the case. So our first speaker. Okay. So we'll have, sorry, Imke, Prof. Nicholas Keswell has been waiting online for quite a while. So we're gonna have him on the first slot. And I'm gonna read his bio. Do you have it? Okay. My co-chair will do that.
Thank you. Please allow me to read Prof. Caswell's bio before he starts the presentation. So where we sit, snakebite envenom is a neglected tropical disease that disproportionately affects rural populations, causing approximately 138,000 deaths annually and leaving many survivors with lifelong disabilities. So Prof. Caswell has taken a multi-pronged approach to increase our biological understanding of snake venom, advancing clinical treatments for snakebite, and influencing global policy. Prof. Caswell is currently the director of the Center for Snakebite Research and Innovations at the Liverpool School of Tropical Medicine, where he leads a team of more than 20 researchers working on tropical snakebite. The center is centered on research activities that are broad, spanning the bench to bedside continuum, and have a strong focus on the discovery and development of new therapeutics to better treat tropical snakebite. So ladies and gentlemen, allow me to give this time to Prof. Caswell so that he shares with us part of what he does. Thank you. Prof. Caswell, please carry on.
Thank you very much. Thank you for that very kind introduction, and apologies for not being with you there today. Hopefully you can hear me okay and see my slides on screen as well. So yeah, as I've just been kind of introduced, I work at the Liverpool School of Tropical Medicine, and my focus predominantly relates around the development of therapeutics for snakebite. And I'm going to give you an overview of some of the work that we've been doing, both in partnership with colleagues at KIPRI and other international collaborators all around the world, to try and mitigate the effect that snakebite has on disadvantaged populations in the tropic. So for those of you perhaps less familiar with snakebite than others, here's an overview of the scale of the burden of snakebite across the world. So we think that perhaps as many as 2.7 million people are envenomed by venomous snakes every year, with the burden of snakebite disproportionately falling upon those found in sub-Saharan Africa and parts of South and Southeast Asia. And collectively, this likely results in over 100,000 deaths and many more cases of long-term morbidity as the result of the toxic action of the venom found in snakes. And I think this image on the right-hand side of the slide that you can see here really encapsulates the great threat of snakebite. So typically agricultural workers working in rural areas of the tropics who are at risk of a snakebite on a daily basis as part of an occupational hazard. Now, although snake bite is a single term and simply encapsulates what happens when people are bitten by venomous snakes, there's a lot of complexity that relates to this issue. And so different venomous snake species have very different toxic molecules within their venom that are introduced when the snake bites. And this causes a great diversity in terms of pathologies in snakebite patients, but it also causes a real challenge when we're developing therapeutics to try and mitigate the toxic effects of these bites. And so here you're seeing a slide that shows you different toxin families that are found in the venom of different snakes, and these are indicated by the acronyms at the bottom of these bar charts. And broadly speaking, there are two medically important families of venomous snakes, the vipers and the elapids, and they have quite distinct toxins in their venom that causes different effects. And so you can see here on the bottom half of this bar chart that the elapid snakes typically have toxins like PLA2s and three-finger toxins that cause neurotoxic effects, whereas the vipers are dominated by enzymes like metalloproteinases and serine proteases that often cause hemorrhage and coagulopathy in snakebite patients. But each venomous snake ultimately has a diversity of different toxins within its venom, and this diversity varies from one snake species to the next, meaning that we have this really quite large pool of different bioactive components that we need to try and neutralize depending upon which snake has bitten a patient. And often, of course, it's not apparent necessarily which species is responsible for a bite, and so our therapies need to be quite broadly effective to encompass the diversity of toxins that are causing harm. So at the moment, we treat snakebite with therapeutics known as antivenoms. These therapies are life-saving if the correct antivenom is selected and given to a patient promptly. And so antivenoms are made by immunizing animals, typically horses or sheep, with small amounts of venom that stimulates an immune response. And we harvest the polyclonal antibodies that are generated by those animals and formulate them as an intravenous therapy that can be delivered to patients after a bite. Although these therapeutics are extremely effective, they have lots of limitations associated with them. They're based on principles that are over 100 years old, so-called serotherapies. They involve their efficacy is ultimately dictated by which venoms are used during the immunization process because of this issue of venom variation. And so products have limited species specificity. So if you were to try and use an antivenom that's been produced against African snake venoms, for example, to treat a bite in South America, it won't be effective because of venom variation. So we have many different products made in different parts of the world with limited geographic reach. And that causes many challenges for the manufacturers in terms of the financial sustainability of these products. Antivenoms also have a poor safety profile because they're animal-derived and then intravenously injected. Many people have adverse reactions to the foreign proteins found in these therapies. We have to manage those adverse reactions in a clinical setting, often in a secondary or tertiary facility. And that means that snakebite patients have to travel often many, many hours or even more than a day to reach a facility that might have antivenom to effect cure. And that's a real challenge because snakebite is a very acute, life-threatening event. And unfortunately, that delay between time of bite and treatment can have a real severe impact on patient outcomes. Other challenges also exist with antivenoms. As I mentioned, that it's a fragmented drug market that's very vulnerable to manufacturers withdrawing products due to a lack of product profitability. And the cost of the treatment itself can be very expensive if this isn't subsidized by a health system, potentially further pushing patients into poverty. And there are also other issues in terms of the regulatory framework guiding antivenom in terms of the quality of the products, whether they're dilute or whether they're concentrated with lots of antibodies, and also how we ensure that inappropriate antivenoms that are produced from one region don't enter a market where they are ultimately going to be ineffective. So recent years, largely driven by funding catalyzed by the Wellcome Trust, there's been a huge interest in developing new therapeutic approaches to snake bites to ultimately, in the long term, replace antivenom therapy. And you can see some examples down the left-hand side of some of the different modalities that have been developed and explored in a preclinical setting. These really include two main topics that I'm going to touch on. One briefly in the case of toxin-specific antibodies, so here monoclonal antibodies in particular, and then secondly small molecule toxin inhibitors, otherwise known as drugs, which I'll focus on for the rest of this talk. So an example of toxin-specific antibodies really comes from a project that we've been working in collaboration with our colleagues at CaseRick at Kitbrae. And this project really has been seeking to use modern technological approaches to identify toxin-specific antibodies that could potentially replace polyclonal antibodies we find in current antivenom. And so we've been immunizing a range of different animals, as well as using naive antibody libraries for monoclonal antibody discovery. Some of these antibodies have different formats that may be more attractive or desirable for snakebite, including things like camelid-derived nanobodies, which are much smaller and able to penetrate tissue more effectively than whole immunoglobulin Gs, and also could potentially be much cheaper to produce recombinantly in the long run as replacement products. And so this collaboration that's ongoing at the moment is really seeking to discover antibodies that are effective against the most medically important snakes found in sub-Saharan Africa and also in India as well. But ultimately, irrespective of which new therapeutic modality we might use as future treatments for snakebite, we have to recognize that the diversity of toxins found in snake venom, particularly when you start comparing across one snake species to another, might mean that we need combinations of different therapeutic molecules to broadly affect cure across diverse snake species. And that's illustrated here in this figure on the scheme. Some toxin families might be more amenable to neutralization by antibodies, whereas others may be more amenable to neutralization by things like small molecule drugs. But ultimately, to provide broad effects across these diverse venom toxins, we're likely going to need multiple therapeutics that we can mix together in a very targeted and specific way to broadly affect cure. So for the rest of this talk, I'm going to talk about the work that we've been doing relating to the discovery and development of small molecule toxin inhibiting drugs, and particularly focusing on the inhibition of a medically important toxin family called the metalloproteinases. So there are many examples where small molecules have been identified in the literature over several decades as inhibiting the enzymes found in snake venom. And drugs offer this unique potential to be broadly effective across an entire class of toxin. Unlike antibodies, they're often very specific to certain toxin isoforms. And that's because these small molecules are able to sit and bind to the active site of enzymes, therefore potentially generically inhibiting their mode of action. Several molecules described in the literature have already been developed for other disease indications and therefore are being explored now for snakebite as a repurposed drug. And that means that many of these drugs have already previously demonstrated safety in clinical trials for other indications, which is really advantageous when you're developing a new therapeutic for a neglected tropical disease because it reduces the time and cost associated with entry into clinical trials. One of the most attractive sides of exploring toxin-inhibiting drugs for snakebite is that there's the potential for oral bioavailability. And so you can envisage a situation where some of these drugs could be taken orally by a snakebite patient in the community very soon after a bite, rather than them having to wait many, many hours until they reach a hospital facility to receive current intravenous antivenom. And there may also be some potential cost benefits as well with these molecules in that chemical synthesis is often much more inexpensive than the production of biologics, whether it be monoclonal antibodies or current conventional antivenom production. And so we've been focusing on metalloproteinases, and I'm going to talk a little bit about the work we've done with a metal chelator known as DMPS as a potential oral therapeutic for snakebite. Metalloproteinases are an enzyme family found in many viper snakes, and they're responsible for causing hemorrhage, coagulopathy, and local tissue damage. And these enzymes rely on zinc for their enzymatic activity. And so several years ago now, we explored the potential of metal chelators as a class of medicines that are already licensed drugs that could be of benefit to generically inhibiting the metalloproteinases found in snake venom. And they do so by essentially capturing the zinc from the active site of these enzymes, rendering them inactive. And so we had a project where we used saw-scale viper venoms, which are very rich in these metalloproteinase toxins, as the basis for our study. So saw-scale vipers are a very medically important group of snakes, have a wide distribution, as you can see on screen, from West Africa through into South Asia. And you can see the venom profiles of this related group of snakes with the blue portion of the pie, the snake venom metalloproteinases. So you can see that for many of these different saw-scale viper species, these metalloproteinases are really the most abundant toxin family found in their venom. And often they are the most predominant pathology causing components too. So our rationale here was if we can use metal chelators to inactivate these metalloproteinases, maybe we have something that could be used as a pre-hospital therapy. And so this work was really led by Laura Albulescu at LSTM with a collaboration with Jeroen Kool in Amsterdam. And what we did in this study, which I don't have time to go through all the details, is we used a variety of different in vitro laboratory assays to explore how well the different metal chelators we were testing inhibited the metalloproteinases before ultimately progressing with our lead chelator, this molecule known as DMPS, into preclinical studies where we explored the ability of the drug to prevent lethality caused by the saw-scale viper in mice. And so our first studies involved a traditional preclinical model of snakebite envenoming where you co-incubate your therapy with your venom, and then you inject this intravenously into a mouse and measure the effect compared to your venom control. And in these survival curves, you can see that the effect of this venom when delivered intravenously causes rapid lethality to the mice within an hour, and that's indicated by the red line on the screen. But when we co-incubated the venom with this metal chelator, you can see that the mice were completely protected for the duration of the experiment. So this was a really nice result showing this concept that perhaps despite the diversity and complexity of snake venom, you can use single molecules to ultimately protect against the lethal effects of certain snake venoms. The limitation with this model is, of course, that in the real world, you're not going to have a situation where your treatment is being co-incubated and co-delivered to the patient that's bitten by a snake. And so after that, we moved to a rescue model in the mice, where here we're injecting the venom intraperitoneally. And again, you can see the rapidity of death in the mice caused by the venom in the red line. So after 4 hours, all the mice have succumbed to the lethal effects of the venom. And then 15 minutes after the venom challenge, we injected the drug. And in the blue line, you can see that we have this really nice window of protection up until about 12 hours after venom challenge before we start to see secondary lethality in the mice. And this is likely due to drug clearance later on. But it provided some support for this premise that, again, you can use a single drug to provide an element of efficacy against envenoming by this species of snake. What was particularly nice was when we combined the drug, early delivery of the drug, with secondary antivenom, trying to mimic what might happen in the real world. So you can imagine a scenario where a patient is bitten by a venomous snake. They're able to receive this metal chelator early, ideally in the community soon after the bite. And then they still go to hospital and receive antivenom secondarily to mop up any remaining toxicity. And you can see in the green line that when we did this in the mice, you can extend that protection initially provided by the drug to 24 hours, which was the end of our experiment. And what I'm not showing you here is the antivenom controls. When you only use antivenom late, so 60 minutes after venom challenge, this isn't fully protective. And so the benefit you're getting from the combination of these two treatments is greater than either DMPS or antivenom alone. But in those studies, we were still injecting the drug, and ultimately, of course, we see the great benefit of these drugs as through their oral use soon after a snake bite. And so our final preclinical experiment involved the oral delivery of the drug after the venom challenge. And we found a similar situation to when we injected it, although there was a reduction in efficacy. So here you're looking at, again, lethality in red from the venom only. In blue, when we use an oral drug after the venom challenge, and you can see there's partial protection with some of the mice surviving and some dying. In purple is the antivenom control, so when the mice received late antivenom an hour later, and again, you can see partial protection with some of the mice surviving. But when we combine the early oral drug delivery with secondary intravenous antivenom, as shown in the green line, you can see that all of the mice survived the experiment without any evidence of severe pathology. And so this really provided us with this evidence for this concept that an oral drug for snake bite that can be given quickly, followed by secondary antivenom in hospital, could be a really important tool to improve patient outcomes. And one of the key things here is that DMPS, also known as Unithiol, is actually a licensed drug under the trade name Demovol. It's already available for use and has been used for many decades for the treatment of heavy metal poisoning. And so this molecule in particular is quite tractable to move into clinical development to further explore its utility as a snakebite treatment. And so that's what we've done in recent years is we've, although DMPS is a licensed drug already, we undertook a phase 1 dose optimization clinical trial in healthy Kenyan volunteers in collaboration with colleagues at the Kemri Wellcome Trust facility in Kilifi. And so at Kemri in Wellcome Trust, they have a clinical trial facility where we were able to enroll healthy volunteers to evaluate the safety of the drug at escalated doses. And the reason we did that is, although Demovol is already a licensed drug, in its treatment of heavy metal poisoning, patients receive a relatively low dose of the drug many times over many, many weeks. Whereas for snakebite, we envisaged patients receiving fewer doses, perhaps only over 1 to 2 days because of the acute nature of snakebite. And our preclinical data suggested that we would need higher doses of this drug than had been tested previously. And so in this study, we escalated the therapeutic dose from 300 milligrams currently used for heavy metal poisoning to 1,500 milligrams. And we were able to show that there was reassuring safety of the drug doses. Even at the highest doses, there was no evidence of any severe adverse reactions. And we were able to show, using pharmacokinetic analysis, that the highest oral dose of the drug gave us a pharmacokinetic profile, or at least peak drug levels, that were highly comparable with an intravenous dose of the drug that is currently used for treating heavy metal poisoning. And then in the second part of this study, we actually moved from single doses of the drug to a multiple dosing regimen where we combined over 24 hours, 3 doses of the drug to see whether we could get high drug levels for a continual period of time that we imagine would be beneficial for snakebite. And again, we were able to show that this combined dose was very well tolerated. There were, again, no severe adverse events. And this provided us with high levels of the drug that were persistent for 48 hours, much higher drug levels than were found with a single oral dose for that period of time. And so ultimately, we believe that this study has provided us with a PK-informed multiple dosing regimen that's now ready for a phase 2 clinical trial evaluation of this drug in real-world snakebite patients. We've recently acquired funding for this study where we're going to evaluate whether DMPS is beneficial for snakebite. And we're going to be performing this study in 2 different places, in Ghana and in Brazil. And we've chosen those two sites because they have snakes that have an abundance of the metalloproteinase toxins in their venom that the drug targets. And our plan is to enroll participants in this trial that have abnormal blood clotting. And we're going to enroll them in the community before they have the opportunity to receive antivenom, which is typically not available in these communities. They'll be randomized to either the oral drug or placebo, and then they'll be transferred to hospital without delay to receive the standard of care, which would also be antivenom. And we're going to use a marker of blood coagulation, the INR, as our primary outcome measure. And our hope is that receiving the drug early, in combined with antivenom, will enable these patients' clotting parameters to return to normal much quicker than they would without the delivery of this oral drug. So just briefly to finish with, I think it's important just to reinforce the fact that ultimately I think we'll need combinations of different molecules. And while the metal chelator I've just described to you is really exciting in terms of its ability to neutralize metalloproteinases, there are many other toxins found in snake venoms that won't be affected by that drug. And so it's important for us to start thinking about how we can combine different inhibitory molecules together to broadly affect cure. And so in this study I'm showing you on screen, Laura took a molecule that inhibits one toxin family, the PLA2, and another inhibitor of the metalloproteinases, and combined them together and tested them against these different vipers found across the world, and that have very different venom compositions. And she was able to show that this combination of two drugs targeting two different toxin families can broadly protect against all of these different snake venoms in the preclinical model, mouse model I described to you before. And I think this provides us with a proof of concept about how we might start developing modular therapies for snakebite moving forwards. So in summary, there's a strong rationale to try and improve snakebite therapies, and there are many different modalities that are under study at the moment, including monoclonal antibodies and small molecule drugs. And ultimately, we might need combinations of different therapeutic molecules to provide broad cure against the diverse types of venom toxins that are available in venomous snake species found across the world. I think the oral bioavailability of drugs in particular may provide us with an additional tool in our arsenal to treat snakebite, perhaps in combination with conventional antivenom or other therapeutic modalities moving forward, but ultimately trying to mitigate that time between bite and treatment that leads to poor patient outcomes at the moment. But there are still really important things for us to address, and certainly designing and funding clinical trials to evaluate these exciting new therapeutics under development is one of the critical next steps for this community. I'll finish there and just say thank you to lots of different collaborators from all over the world, including colleagues such as George and his team at CaseRick in Kipre, and of course the funders who support our work too. Thank you.
Thank you very much, Prof. Caswell, for that wonderful presentation on snake bites and, of course, what you're doing to help us avoid that. In the interest of time, I'll just allow 2 quick questions for Prof. Caswell.
Thank you very much, Prof, for your presentation. I'm Dennis Omara from Uganda Virus Research Institute. I have only 2 questions. First, on the animals that you are using to generate the antibodies against the venom, you cited the horses. Why these animals, not any other animals? And if there is any other alternative you could elaborate, or any improvement in terms of the animals. And the second one, convalescent plasma therapy is one of the immunotherapies that have been explored for treatment of disease post getting the antigen in the body. Do you think this could also be explored since we have snake bite survivors among us? Do you think this option could also be explored for generating the antibody? Thank you.
Thank you. Yeah, great questions. I think the use of equines and horses is largely a historical for historical regions. So many years ago, when antivenoms were being developed, we noticed that horse antibodies can be separated from plasma via sedimentation and didn't require centrifugation, and so this was an efficient way to isolate the antibodies. Horses are also quite attractive in that they have a large blood volume, and so the cost of maintaining horses is relatively inexpensive compared to other potential animals. But in theory, you could use any animal that can produce antibodies for this process, and so people have explored using camelids, they've explored using chickens, they have explored using sheep, and there are products on the market derived from sheep too. So there isn't a very strong rationale to necessarily only use horses, but this has become convention. And then your second point around survivors is a really valid one. And in fact, there are people who are working on antibodies that have been isolated from people who have been bitten by venomous snakes. And there are publications of some very interesting monoclonal antibodies that appear to be very potent, that have been isolated by individuals who have been bitten by snakes several times as well. So this is an ethically challenging area to work in, compared to using naive human antibody libraries. But there is clearly a strong rationale in that these individuals may have developed antibodies with higher affinities to venom toxins than those people who haven't been exposed to them.
Thank you very much, Prof. My friend is well guided. We'll take just one more last question.
I have a question. For the phase 2A trial, will the participants be placed on anticoagulants?
No, they won't be placed on anticoagulants. These are often contraindicated in the case of snake bite that causes coagulopathy. There's a risk there that it may exacerbate the existing coagulopathy, so the patients won't receive any anticoagulant therapy as part of the trial.
Thank you very much, Prof. Caswell. We really appreciate your effort and, of course, everything you put in together towards this talk. In the event of any more questions, we're happy to collect them and present them to Prof. Caswell via email. I think his email contact is also shared in the abstract book. So if you're eager to interact with him, please go ahead. And at this point, let's please applaud Prof. Caswell for the work well done.
Thank you.
Great. We're now moving on to the next presenter.
Welcome on stage Prof. Yemkast Iniestra. As she comes, I'll introduce and give a brief bio of her. Prof. Yemkast Iniestra is a professor of medicine, internist, infectious diseases consultant, and clinical epidemiologist with 22 years of clinical and public health research expertise in NTDs. Her research experience includes both quantitative and qualitative methodology, implementation research, and she's undertaken numerous rapid RCTs, including in Buruli ulcer, in Ghana and Benin. These trials, in close collaboration with the WHO, have changed both the antimicrobial and surgical management of Buruli ulcer worldwide. I do work with her as one of my co-PIs and directors at the African Snakebite Alliance. Welcome. And before she begins, I'm told to remind you that there's a parallel session on climate health and ecology happening in the Rex room, so those interested could walk to that session. Karibu.
About snake bites. Could you hear me? Or? Okay. Well, Georgia said this was, you did not quite like this time spot. I do, because early in the morning, you still have to wake up. After lunch, you gradually fall asleep, and I hope you're not waking up again. So I think this is the best time slot for neglected tropical diseases, and I hope the 21st conference will again be this time slot. I'm going to talk about snakes, and I realize there are people who are really afraid of snakes. And maybe then you are in room B. But otherwise, I just show you this slide to show you that it is completely normal that for people to be afraid of snakes. And these photos were shown to very young children, actually toddlers. And they looked at their response to these photos, comparing how they responded to the orange goldfish and the orange-colored snake. And based on their responses, they concluded that the fear of snakes is something inborn. Now, methodologically, there were some limitations, but I want to keep this slide, so I will not discuss these limitations. Main conclusion, fear of snakes is perfectly fine and normal. Why snake bites? It is the deadliest neglected tropical disease. Now, it's not a competition, but yet it is relevant because despite being the deadliest and having quite a lot of long-term consequences, it's been acknowledged as a neglected tropical disease in 2017. So that's fairly recent, especially if you realize the snake have been around for a very long time. So the estimate of death in this study would be more than 60,000 per year and almost 3 million years of life lost. What I want to emphasize is that the different snake species we have lead to different challenges to our healthcare systems and different clinical syndromes in our patients. So on your left, you have the black mamba, which would lead to neurotoxicity, meaning you would have to go to the hospital as quickly as possible. So that kind of healthcare system you would really need a good ambulance system to make it to a hospital as soon as possible, hoping that there are intensive care facilities there in case you need intubation. In the middle, you see a soft-skilled viper, which would lead to bleeding disorders, Professor Casewell discussed. So that is a very different type of clinical care to provide. And you see to the right is a puff adder. A lot of cytotoxicity requiring long-term follow-up, wound care, maybe surgery, skin grafting. So the different snakes lead to different problems and different challenges to your healthcare system. Now, despite these snakes being there for such a long time, and the mortality and morbidity, same thing, we still have very, very, very big evidence gaps in the clinical and public health space. And in this figure, we looked at publications with at least one scientist based in Africa on the publication, but on snake bites. And what you can see that there are quite some publications, there are not too many, but the ones there are on conservation and on the venom. But on how to treat, the epidemiology, the long-term impact, there's almost nothing there. So one of the initiatives to try and improve that evidence gap was to start the African Snakebite Alliance, which I co-lead with George Olioch, but probably because he's chairing, I'm the one now speaking. And it was launched here at Kipre, as you can see. And what is its aim? Its aim is to improve the health outcome for people affected by snakebite envenoming by addressing evidence gaps in policy and practice. With 3 objectives: to undertake high-quality research, support evidence-informed decision-making in policy and practice, and to develop and support a sustainable and cross-disciplinary capacity for snakebite envenoming. So I already mentioned that there was so many unknowns. So now I'd like to go through some of these unknowns with you and discuss what kind of research the African snakebite is currently doing or going to initiate for you to have an idea on the kind of things that we are up to. So imagine you are the person, no, not you. Imagine someone who gets bitten, and the left photo is a field with cassava seedlings, and then you get bitten, and what do you do next? Who gets bitten and where and how many people is one of the things we have very, very poor information on. Where should people go to first? How do they make the decisions? Do they first go to the community, to the CHPs, or to the traditional healers before going to the emergency room? How to treat snakebite envenoming, Prof. Caswell elaborated on that treatment, but there are other aspects of that treatment that we do not know currently. And what happens after the bite? So why was I saying we don't really know how many people get bitten and where? It's even though we have this slide and it has a number, more than 63,000 deaths, but there are other publications which you will, if you look for them, will report different numbers. And why does that happen? Well, you can see on the left side is one study, and that study is from, I think, 2008. And the light brown countries, and you can see it's many of the light brown countries are in Africa, there are no numbers. What that means that the studies we found extrapolated data from the very limited available data and kind of used that to make the estimates for the entire continent, which you can, as you can imagine, has quite some limitations. That was 2008. But then the study at the right is the one I presented first, was from 2022, if I'm correct. And they had also their estimates, but still you can see the gray countries in Africa where there's no data at all. So again, extrapolation. So that's why you find these very different numbers in literature. And any lecture you attend on snakebite may have different numbers for that reason. So the who and where has very poor data. And I see some of my keeper colleagues sitting over there. This is one of the research we are currently working on. What the keeper team did is to link up with mass drug administration initiatives. And following that strategy, more than 13 million Kenyan inhabitants were asked if they were bitten by a snake in the past year. So what happens if you do a community survey? You go to a community where you know snakebite is a problem, because with the limited funding you have, that's where you will start. But by linking up with the mass drug administration, you can actually see that the snakebite problem varies enormously within Kenya, with the dark red, brown, almost black colored being the having the very, very high incidences. So that will dramatically improve at least the snakebite data you have in Kenya. And it's also a message to other countries, because there are, of course, many countries where mass drug administrations are being given. So I said the numbers to differ. How much? Well, you can see in Baringo, I call it the highest scoring countries, red and the lowest as well. Baringo and Turkana had like more than 400 cases per 100,000 people per year. And then you can see the lowest numbers are between 3.5 and 4. Very different. And that can help you planning your educational interventions, or maybe planning your antivenom distribution. You need that kind of data. Okay, so if you have better data, the next step is where do people go to? If they are bitten, how can we support them getting their access to care? So one of the research initiatives that in the African Snakebite Alliance that will start soon will be done by Rita Salifu from Ghana. And her work will be on women's livelihoods in rural communities near Mole National Park. She will focus on women who work in the shea butter, as a trade in shea butter. And she will then look at how to guide policy to protect these at-risk women and strengthen the local health systems. So trying to enable them to get quicker access to care if needed. Again, the Keeper team is looking at beekeepers in Baringo and see what kind of risks they have and why the beekeepers, because these hives went up in the trees, is also where black mambas like to hang out. And they like the hives, probably for temperature reason, or maybe because there are animals moving towards these hives to get the honey that are interesting for the snakes. But you can imagine as a beekeeper that has quite some risks if you are standing in the trees and working with your hives. So we're not only trying to find out how often this happens, but mainly want to focus on how we can empower these beekeepers to prevent getting bitten and to support them in what to do if they get bitten. And how to treat. Prof. Casewell listed quite some options we currently have, or we will hopefully have in the future. So Robert Asampong from Ghana looked at the access to care and the time to care, actually. Because you can have the best treatment ever, but if people take hours and hours to get there, you still have a problem. So on the left, you can see what happens if people have to walk in Ghana to get to a hospital for care. And the gray color indicates it's more than 4 hours. So you would want to be at least, depending on the type of bite and clinical problems you have, but definitely you want to be there within 4 hours. Now snake bites, the majority of the bites happen in the north. And you can see that there is an awful lot of gray. An awful lot of gray. Of course, it looks a bit better if you start walking and then find a motorized vehicle, but these figures still look problematic. And then what happens if your treatment moves from the big hospitals to the health centers and clinics? Then you can see that the time to care is really improving. We're not there yet, but it does get better. And at the right, you can see what happens if the treatment is also available at the health posts. So in that case, access dramatically improves. And that is linked, if Prof. Casewell mentioned, if you would have oral treatment available in communities, then you would dramatically improve time to care. Talking about treatment, you also have strategies to be able to evaluate the intervention you have, looking at infrared imaging and ways to assess the long-term consequences, because you can have a good new treatment option, but you also need ways to evaluate its impact. Prevention is another very important aspect, because if you prevent bite, you don't need treatment at all. And what happens after the bite is something else our colleagues from Keeper did. We looked at the disabilities in patients after the bite and compared snakebite survivors with community controls. So, summarizing, this study shows that avoiding death in snake bites is very cheap. And they looked at how the cost per death avoided and the cost per DALI avoided. And they focused mainly on antivenom. And it's less expensive than other neglected tropical disease to reduce both death and burden. And this is the numbers you have if you look at antivenom only. Now, if we also look at prevention, improving time to care, availability of care, and improving treatment protocols, these numbers, saving lives, would be even cheaper. So I would like to close with listing all core members of the African Snakebite Alliance, and I hope you now have a clear idea on the kind of research and activities we are undertaking. Thank you very much for listening.
Thank you, Prof. We can give one more round of applause. We just have time for one very burning question. Just one very burning question. Okay, you raised your hand first.
Good presentation. My question is, for the snake bite survivors, are they eligible to donate blood?
Eligible to?
To donate blood to a recipient who has not been exposed.
Sorry, I'm not quite. You mean if they can donate blood in case?
Okay. Afterwards, maybe throughout their lives, are they eligible to donate blood to a recipient that has not been exposed to snake bites before?
Yeah, there is no reason why a snakebite survivor could not be a blood donor for other medical indications of receiving blood. Any other questions? No? Well, thank you very much for listening.
Thank you very much, Prof, for that invigorating presentation on snake bites. Ladies and gentlemen, we're moving on to the next presenter, who's none other than Dr. George Omondi, and allow me to read his brief bio. So Dr. George Omondi is a senior veterinary surgeon and a tropical and infectious disease research scientist who leads innovative programs that prevent and control tropical infections. And he is interested in addressing snake bites and the global burden in African communities. And his research focuses on new therapeutics, community interventions with health policy, advocacy, and fundraising. He is very passionate about leveraging science for Africa's development, and he fosters partnerships among African institutions, countries, and international donors through securing major grants from funders like the National Research Fund of Kenya, the Royal Society of the United Kingdom, the Wellcome Trust, and many others. He has authored over 20 publications and serves on the Kenya Ministry of Health Snakebite Technical Advisory Group. So, Dr. George, you're most welcome to take us through your presentation.
Good afternoon. A lot of what I was going to talk about has already been touched on by my colleagues and partners. So for the sake of time, I'll want to share what we really do at KSRIC, the Kenya Snakebite Research and Intervention Center, towards accelerating science policy and equity for snakebite. So many diverse contributions in the research space. Kenya, we suffer at least 20,000 snakebite cases every year. This is a gross underestimation. And like Prof. Imka mentioned earlier, we are working towards getting out the right estimates. We think that around 1,000 people die in Kenya. And again, this is underreported, and the psychological burden, which is quite pronounced, is yet to be determined. In the Kenyan space, again, mostly pastoralists, farmers, children in arid and semi-arid regions suffer the greatest burden. And we do know, like has been mentioned by Professor Nick Caswell, that antivenom is the only specific therapy. That global burden map has already been shared by Professor Nick, so I'm not going to dwell on it. So what is really eye-catching about Africa or sub-Saharan Africa is that the case fatality rate as compared to other regions where snakes do exist and also do cause morbidity and mortality, it's quite high at 7.6 as compared to the rest of the world. So why is this so? We do know that we have at least 400 species of snakes in Africa, 30 of them which are of medical importance, and these 30 species of medical importance have quite a wide variety of venom protein or toxin composition, some of which actually are intraspecies. You also do find some geographical variation as well. These venom toxins do, of course, lead to various diverse local tissue issues and lethal systemic effects. And we do know that snakebite is a medical emergency requiring rapid hospital-based treatment with antivenom. Prof. Imka alluded to the fact that it's the most expensive NTD to treat. It does affect impoverished populations in rural and remote communities whose health infrastructure are mostly under-resourced, of course, financially and clinically, with limited trained staff to treat snakebite or snakebite envenoming, and antivenom is mostly also, of course, in short supply. So we should be able to treat these venom-induced systemic pathologies that we've talked about and Prof. Caswell talked about earlier, and of course without the aid of diagnostics. So our medical care workers in these areas rely on very rudimentary ways of diagnosing snakebite, and even if they do, they have to again deal with antivenom-induced adverse effects without sometimes the aid of oxygen, and this has been shown to lead to deaths. And then in Kenya, we do again suffer mostly venom-induced tissue necrosis. So reducing the disease burden requires context-appropriate research addressing these diverse barriers to healthy outcomes. So at KIPRE, which is hosting the Kenya Snakebite Research and Intervention Center, we did establish the unit covering these various research areas, so public health, venom studies, clinical research, and research into next generation snakebite therapies, and a lot of capacity strengthening and advocacy, because when we got into this area, we realized that a lot of scientists who were well established in other areas like malaria, TB, HIV, were not really aware of the issues in the snakebite area. So we've done a lot of advocacy to bring people on board, sensitization, etc., and bringing in multidisciplinary expertise to enable us answer some of the research questions. So we are working towards establishing Kenya and regional venom reference standards. Prof. Nick Caswell talked about the composition of venom, and they're secreted from specialized glands in the upper jaw of the snakes. So when a snake does bite, they either inject venom if they're venomous, and at times we talk about dry bites if you feel that they don't really need to kill you, a human, and later have you as prey. And again, this has been, we've talked about it varying between species, region, and sometimes age groups of the snakes. The proteomics, transcriptomics, and functional assays for characterization, we are currently undertaking a lot of this to establish the venom reference standards for Kenya and the East African region. Caswell talked about the marine models that we're using for toxicity, and again, just improving the animal welfare issues around this space. We're also coming up with some in vitro techniques to better understand the the toxicity of venoms. So we'll be hoping that this informs better and better region-specific antivenoms that we are currently working towards producing. So this has also been touched on. The conventional and historical way of producing antivenom is having sublethal venom doses injected into a horse. We have various other animal models that have been used collecting the plasma, or some people serum, and purifying the IgG. Some are now breaking that down to the Fab prime and packaging that as antivenom. We have newer techniques, what we're terming as the next generation snakebite therapies, and we're working on our monoclonal antibodies for that. Prof. Nick Caswell talked about that. Of key importance is that a lot of the antivenoms over the years that have been coming into the sub-Saharan African region have been coming without any preclinical or clinical efficacy and safety data. So we're working very hard to establish a regional quality control unit for testing batch by batch antivenoms coming into the region. And we look to really improve on this, as shown in this publication that we did in 2017, where we found some of the antivenoms are actually basically water, because manufacturers, when they know someone is not checking, then they try and play some of those dirty games. So as we try and establish equity, because we talked about NTDs affecting mostly the marginalized, and we do think that snakebite does affect the poor of the poorest amongst the NTDs community. And we have talked about antivenoms being expensive. We also do know that they're mostly unaffordable because of, the historical 200, it's a historical 200 years of it not being changed from the initial when it was discovered. So it has become, it is expensive, and that leads to its unavailability. And also because of the neglect, we've had issues of its ineffectivity, and this, of course, does cause equity. We've had market failures from big farmers, so they've stopped production over the years, and we are trying to change that through local production. The cold chain limitations, areas without electricity, so not being able to be stored appropriately. And we looked that in the next 2 years, we're going to be having some of the first batches of vials that are produced locally. We've invested heavily on snake mapping to be able to establish hotspot areas and provide real-time data on which species are available where. This is very useful for us in that it does enable the healthcare practitioners to narrow down on their diagnosis. And that's a map of Marsabit and showing the snakes that are available in that area that we've been able to map using GIS, Kilifi as well, Kakamega and Busia. We've also been able to produce those maps, Baringo as well, and a few other counties. So our snakebite clinical research, what are we trying to achieve here? Of course, we are setting up for clinical trials and efficacy studies. We are also establishing snakebite diagnostics tools, and at the same time trying to understand the pathophysiology of snakebite envenoming. The current diagnostic tools for snakebite envenomation really rely on the syndromic approach, identification of dead snakes or photographed snakes, which is sometimes risky because the patient or whoever is around could also be bitten. And novel methods that we are working towards answering this is that we've currently sequenced 6 species. So these 6 species of snakes we've we've gotten back data on their whole genome sequences. The de novo assembly is currently being undertaken by a team led by Dr. Mercy Ekinyi, and we hope to publish this. This, in the past, has limited people who are trying to work on the diagnostic space, and of course, with this new data, that will make it easy for them. We are currently also undertaking studies to assess the biomarkers over time that will predict the disease progression and guide treatment, and we're collaborating with health facilities in Kitui, Kilifi, Baringo, Samburu, and Turkana. We're also coming up with other novel ways of diagnosing snake bites. So with Profimca and the team, we are currently investigating the point-of-care infrared thermography as a diagnostic tool to assess the progression of local symptoms of envenoming. And the potential role of this is considering that a lot of the bites in our area present with sector toxicity, we're able to show the different variants and patterns over time with the infrared. We're able to also explore new tools to evaluate patients with symptoms of cytotoxicity, envenoming, and local tissue damage. And as we collect the blood samples, we're also testing for novel molecular techniques for ID of this of the snake species in these patients. So again, this study is happening in Kitui, and just using the sandwich ELISA, we're able to again show which snakes are available. Yep, okay, I'm summarizing. So we also been able to determine which of the, some of the preliminary data, which areas of the body the snakes mostly bite, and which snakes are mostly biting people in Kitui. And for that region, we can see the puffad and the red spitters are mostly involved. Public health research tries to produce evidence-based data towards really informing policy. So the current situation is that the burden data that we have are grossly underestimating the numbers. We suffer incomplete record in our healthcare facilities. The surveillance data from the ministry, again, is not accurate. We don't know which areas that we, and again, this is leading to not knowing which areas that we will properly, KEMSA will stock the antivenoms. So we've gone on to map snakebite endemic counties. We've already completed 24 counties, and some of that is preliminary data. Kitui, we're seeing an incidence of 270, which is actually quite high. Some regions like Kajiado, 20. Again, this variable data, like Profimca mentioned earlier, is really important in guiding the authorities on which places to stock antivenom. MDA, Profimca didn't talk about that. And we're looking at Turkana, that incidence is quite high. And again, the intervention should be tailored towards addressing that. I've talked about the health facility data, Kenya Health Information System. We found it very wanting, and so this study is going to, of course, address how to improve the reporting of snakebite at the health facility level. We are working with community health promoters through the electronic community health information system to report snakebite and also the functional disabilities over time. And through the mobility assessments, we've been able to determine the functional limitations in a number of counties. And again, this will advise rehabilitation programs of either having snakebite patients with prosthetics or those who have PTSD, and how they can be psychologically treated so that they can become full members of the society again. Pictures of a few of the snakebite victims that we've been able to map out. We've also been able to assess the livestock mortality of livestock from snake bites, and a number of those counties have been assessed. And of course, compared to other diseases, drought, floods, snakebite actually does cause considerable deaths in livestock. And does this warrant us having a snakebite antivenom formulation for just livestock? Because the human formulation, again, is quite expensive. Costa Rica has been able to do that. We've been able to assess the training needs of healthcare workers and identify the skills that they're lacking, and again, this will advise us to the training programs. So I'll stop there, and thank you very much for listening.
Thank you very much, Dr. Omondi, for that presentation on the global burden of snake bites. In the interest of time, I'll just have one question, a pressing one for Dr. Omondi. Anyone with a pressing question? Great.
Thank you for a wonderful presentation. This question was to go to Professor Kessel, but I've realized that you're suffering from the same disease. When it comes to anti-venom production, you have said that you normally use antibodies. So can you bring more light when it comes to broad spectrum anti-venom? Because I know that based on immunology, antibodies are specific. But now, how do you develop broad spectrum or general antivenom?
Would you like me to try and answer that question? I think he said to myself, is that right?
Oh, yes, please, go ahead.
Yeah, no, it's a great question. Most people in the field at the moment, as you say, antibodies are very specific to certain molecules. And so many scientists at the moment are trying to essentially engineer their specific antibodies to be much broader than would be normal. And so this can be done in a variety of different ways. What is done most commonly is when you're selecting your antibodies, you use a specific toxin as the bait to find that antibody. But then you can do another search using a different toxin and a different toxin until your pool of antibodies gets narrower, but you're left with specific antibodies that can broadly interact with those different toxins that you've presented them with. And so there are ways then that you can also engineer the antibodies to bind with higher affinity than they would have done before too. But it's a real challenge, and I think this is one of the reasons why polyclonal antivenoms work very well. They have broad antibodies that interact with lots of different toxins, and so now people are trying to use monoclonal antibodies, which have many benefits, but they also have a weakness in that they're limited to being very specific to certain toxins, and we have to engineer them to bring that breadth back so we can neutralize different toxins at the same time. But it can be done, and there are a few good examples that are in the literature from recent times where individuals have done this quite successfully against certain types of toxin family.
Thank you very much, Prof. Caswell, for taking that question. We really appreciate, and my friend from this end is well guided. Ladies and gentlemen, we're moving on to the next session. It's going to be unique because it's a session that is co-shared, by two speakers. We'll have Dr. Paul Mireji and Professor Richard Ichodi speaking to us. And please allow me to read their bios. I'll start with Dr. Paul Mireji's bio. Dr. Paul Mireji is a senior research scientist at the Biotechnology Research Institute of the Kenya Agricultural and Livestock Research Organization. He specializes in entomology, molecular biology, genetics, and bioinformatics with focus on the physiology, ecology, and the control of vectors and vector-borne diseases in Eastern Africa. He particularly centers on seseflies and mosquitoes, and his work spans graduate studies on mosquito adaptations to urban pollution, His postdoctoral studies were on flies and mosquito biology and pathogens. And currently he has ongoing work on laboratory investigations using molecular tools and high throughput analysis, complemented by his roles as a guest lecturer at the London School of Tropical Hygiene and Medicine. And he's also an academic editor for several journals. He organizes a couple of regional workshops and is a contact for the International Union of Biochemistry and Molecular Biology within Kenya. So please allow me to then proceed to read the bio for Professor Richard Echodi, who is going to share the same presentation with okay, great. I'll read Professor Richard Echodi's bio, who's going to share the same session with Dr. Paul Mireji. So Professor Richard Echodi is the director of the Gulu University Multifunctional Research Laboratory that is in Uganda, and his interests are in epidemiology and population genetics with focus on genomes and the diagnosis of tropical diseases. He's a beneficiary of the Fogarty Global Infectious Diseases Training for his PhD at the Yale University in the United States. So these two will share a session of just about 12 minutes. Dr. please strictly stick to the time.
Thank you. Thank you very much for this, that introduction. We are shifting gears here a little bit. I'm going to talk about annotation, the annotations that we've done on improving the annotation. Hello? Yeah, so we are now shifting gears a little bit back to another beast again that feeds exclusively on blood. So get started by talking about what we've done, the lessons we've learned, and the next steps. So tsetse flies are obligate blood feeders. They are adenotropic, viviparous insects. They're cell biological vectors of sleeping sickness, and being exclusively present in sub-Saharan Africa, they don't diapause, and both male and female are actually vectors of trypanosomiasis. When you look at the arthropoda, that's where we have the insects and ticks and everybody else. Hematophagy has actually emerged 21 times. Among them, we have the tsetse fly that I've highlighted there in red. And actually, phylogenetically, tsetse flies are much closer to Drosophila, which has actually been very important in helping us in annotation of the fly genome. So tsetse flies actually come in 3 different groups. We have the ecologically, we have the savanna species, riverine species, and of course the forest type, and they all have different bionomics and in disease transmission. And we have the most extant population is actually the forest type as shown by the graph below. So tsetse fly also have very significant differences or differentials in terms of vector competence. And coming out of these, others actually transmit the human form of trypanosomiasis, while all of them transmit the aminal form. And at the same time, they also get attracted to different hosts at different times, and visually, they also respond to different colors. So that information informed the initial white paper written by my colleagues at Yale University. And most of it came out of a need to do a comparative analysis of the genomes of all these tsetse flies, so that we could be able to understand the molecular basis of these differential behaviors that have been observed in the tsetse fly. And one of the things that came out was a need to know whether the differences in things to do with vector competence, hematophagy, why do they feed on blood, and why do we have do they have differential discrimination of the different hosts? And of course, they also need to do SNP analysis to look at what are some of the bases that drive some of those differences. So This led to funding by NIH to do a comparative analysis of these genomes. And when this came out, one of the goals that we expected was that we would be able to have an understanding of why and the processes that underline those differences. So when we first sequenced the genome, the first genome we did was the Mositans group. That was just one of the groups. And from that publication, we were able to identify some very important factors. One of the most important things that came out was a reduction in gene copies and numbers compared to what had been seen in Drosophila. And so that being the primary goal of the research being to identify some of the molecular basis of the genes that confer these differences in phenotypes that I've just described that could then be exploited to identify other tools that could be used to expand control efforts. Unfortunately, we did not see any difference when you compare the genome, so that actually raised our curiosity. And that's now where my student who came in my lab, for a master's student, took over. And the first thing she noticed is that from all the papers that we published, trying to compare this, looking at the RNA-seq profile for all the tissues that came out based on the genome, that most of the genomes, some of the reads were actually unmapped, and so we suspect that could actually be representing some of the genes that had not been annotated. And so to prove that, she went ahead and did a de novo assembly of a mapped read, mapped them back, and realized that most of them were mapping back. The de novo found actually around 41% of the antennally expressed genes were not mapping on the transcript, but were mapping on the genome. And when she mapped them back, she found actually a good number, 74% of them were actually novel. So we felt that that could actually be explaining the differences that we had seen. And so when she went ahead and looked at this and found that out of these genes, when she annotated them, she actually identified 592 genes. Among 202 of them were novel, and only 94 of them had actually orthologs in Drosophila and related insects, and 22, around 88 of them had orthologs in Uniprot, and of course 8 were unique to Drosophila. So coming out of these, she also did some analysis and established that most of the genes here were actually associated with repellence or attraction or feeding or non-feeding in tsetse flies. So coming out of this information, and most of the genes actually she also identified were actually associated with, some of them were synthesis, transcription, most of them were not actually associated directly with olfaction, but were associated with other factors in the antennal regulatory process. So what came out of this, the major lesson that we learned out of this is that every genome we look at should actually be treated more as a draft genome, and we cannot be able, until we prove using RNA-seq evidence that all the genomes have been annotated, that we can be able to probably move to the next step and consider any outcome of analysis as something valid. So at this point, I'll invite my colleague and friend, Professor Chodu, to come and talk about the next aspect of the study based in Uganda on population genetics of tsetse flies. Thank you so much. I did mention right at the beginning, this work was done by an MSc student. She has now moved over to University of Nevada in Reno in the US to continue with her PhD program. So she takes all the credit for this work.
Okay, thank you very much, Paul. I'm grateful for the opportunity. I'm Richard Echodu, a professor at Gulu University in Uganda. As Paul stated, this work builds up on much of the work that has been going on within the East Africa. And we are going to give an example where we can use some of this information to guide controls in our different. It's not only going to be on tsetse, but during the course of the week, we have talked about leishmania, cysto, malaria, and many other things. So you could borrow a lift from this and see how we can be able to use this information for purposes of informing control. I will just give a snapshot of my institution. Today we are in Nairobi National Park. So next we will call you at the source of the Nile in Uganda. That basically, before you go to my place, you will be at this facility. This is basically a research center, focused into a couple of things. As I said, more tropical diseases, infectious, and many, many other things. We have over 30 labs. So welcome you guys to come to Uganda. During COVID, to readjust, and the photos you see down here, we had around the clock, and we never lost any student or any of our staff due to COVID. So we had to repurpose the lab from trypanosomes, malaria, and we handled that. So see the change on that. But importantly, we are located between DRC and South Sudan. So we present unique dynamics for you guys who are interested in diseases. So you better come to Uganda. What guides us is WHO together with African context. WHO is focusing eliminating some of this, including sleeping sickness, but the approach is to cut the transmission. Whereas within the African Union, we are thinking of eliminating tsetse flies. So those are two distinct approaches where we need to see how we can be able to contribute in addressing that. So for population genetics, for example, in Uganda, how are you going to handle this? Are you just going to spread spraying control traps everywhere? So with the population genetics, you are able to pinpoint and direct control at what scale, at what disposal should be able to do that. And it's very, very important to use some of this on guiding control. But the biggest challenge in most of these contexts, given the dynamics within the region, bias into South Sudan, DRC, we have seen the resurgence, much as TB Gambia, it's not a public health problem now. But we are still having challenges because of this emergence of like COVID, Ebola, and mostly instability within the region, and that's a cause of alarm for us. So using some of the data that Paul talked about, the genomics, we managed to get the SNPs, microsets, and basically doing sequencing to inform what's going on. There are around 4 things here, is that most of these animals still act as reservoirs, and you can be able to that are coming from the animal. But above all, you can go and pinpoint in terms of the population dynamics where some of these parasites are coming from. So issues of reservoirs are very, very important in the transmission. But above all, we have also seen that the vectors really expand the transmission along very many areas. For the Pepe Tsetse, in Uganda, we are I think one of the few African countries who have that detailed analysis of each and every part within the country, from the lake to Lake Kyoga and the northern one. So we find that most of these Tsetse populations are interconnected. So if you are talking issues of control, you need to be mindful of reinvasion, but above all, how we can be able to guide the national control program. National control program, so that we can do a roller carpet. What we see is that the Tsetse are very interconnected in terms of the water bodies, and that is a cause of alarm, especially if you are going to do control in some of those areas that we are seeing. And the populations overlap within the East African region, including Kenya, Tanzania, and in that condition, if these countries have to work together, they need to jointly make sure that they can be able to put resources together, because within the Lake Victoria region, there's a lot of interconnectivity within the Tsetse population, and that basically means that Uganda cannot do it alone. There is need to work together on that. Because of that, how do we visualize going forward, especially for the young people, with the advent of artificial intelligence, GIS, we are seeing drones planting trees, and the target of WHO is use tiny traps to do the control. So with guidance or with population genetics, together genomics, and these innovations that are coming up front, we feel we are at the right position to guide in terms of elimination of GAMBS, a form of sleeping sickness in the country. With that, I thank you very much for listening to me.
Thank you, Prof. We have time for just one burning question because we're way behind time. Any? If not, then we'll proceed for the next session, and we can give Prof and his colleague another round of applause. Thank you.
Thank you so much, Dr. George and Dr. Kitui, for the session. I think at this time, we will allow for a 5-minute break so that we can exchange rooms for those who want to exchange rooms. There are people in room B. There are presenters in room B who are waiting for an audience on drug development and AMR mitigation strategies. It's going to be a very exciting talk. So let's use these 5 minutes to transition to the next room. And those who are remaining here, remaining on genomics, bioinformatics, and AI in biomedical research. So let's give that time for an exchange. Just next door for those who are doing drug development and AMR. Okay, our 5 minutes are always up, almost up. The session is chaired by Dr. Tunga and Dr. Stella Irungu next door, and the session here is chaired by myself and Dr. Clement Shihuli. Dr. Clement. Okay, so I think we've given sufficient time for people to transition to the other room, those who are interested in drug development and AMR. So in this session, we have a list of very exciting speakers. I know it's coming towards the end of the day, but I promise you it will be exciting. So it's an honor and pleasure to invite Professor Isabella Oyer to come and give the keynote for this session. Professor Isabella is the head of the Biosciences Department at Kemri Wellcome Trust Research Program, and she's a professor of molecular epidemiology, Newfield Department of Medicine, University of Oxford, a Callista Juma Fellow, funded by the Bill and Melinda Gates Foundation, and a Global Research Fellow at the Reuben College, University of Oxford. Her current research focuses on integrating malaria molecular epidemiology into routine surveillance in Kenya, a project that partners with the Division of National Malaria Program to implement malaria molecular surveillance activities. During the COVID-19 pandemic, she led the COVID-19 testing for the coastal region. Kemri Wellcome Trust Program was a regional COVID-19 genomic surveillance reference lab for Africa CDC and WHO Afro, and she led and coordinated this effort. In addition, she is coordinating the scale-up COVID-19 immunological surveillance in the East African region to determine genetic variants with immune escape potential. More recently, she is co-leading a Wellcome-funded 4 pathogens genomic surveillance grant that cuts across 8 countries: Cambodia, Indonesia, Kenya, Laos, Malawi, South Africa, Thailand, and Vietnam. And with that, I welcome Professor Isabella. Karibu sana.
Yep, thank you very much. Merci. Okay, great. Just make sure I know where it is. Okay, great. Thank you. So I'm going to tell you about the work that I'm doing to advance malaria molecular surveillance and for control through molecular innovation. So I start with a news clip that came out in July last year, urging the globe, urging us in Africa about the problem of anti-malarial drug resistance. So they were highlighting that resistance has emerged in Africa. And the last time this happened, it was quite debilitating in terms of the number of deaths due to malaria in children had tripled. So that was an alarming note to the globe to respond. So we have a couple of issues with malaria at the moment. We've had a glorious period in the 2000 to about 2015 where we had a decline in malaria cases, and we could see malaria deaths and malaria cases had gone down. In the last sort of 10 years, we've flatlined, and we've not been able to have an impact on the reduction in malaria cases or malaria deaths. And so what's brewing in the region and what's brewing globally is we have what we're calling biological threats, where the malaria parasite is evading diagnostics, the mosquitoes are evading insecticides, treatment is a challenge. When you go to a health facility and you have malaria, the malaria parasite is escaping treatment, and we have the invasion of new malaria vector species, and this is particularly in Africa, where we never had Anopheles stevensii, but in the Horn of Africa and East Africa, it has been detected as well as in West Africa. So what does that mean when we're thinking about malaria control? We have a conundrum of problems that we'd have to consider when we're controlling malaria. But I'm only going to talk about one of them, and that's about malaria treatment. So historically, we've had to change anti-malarial treatment strategies at national level and at global level. We initially used to use chloroquine up until the late 90s. Between the late 90s and early 2000s, we shifted to sulfadoxine pyrimethamine because the chloroquine was failing. People were going to the hospital, taking the 7-day treatment of chloroquine and not resolving their fevers. And some of these individuals ended up having severe malaria. In mid-2000s, we switched to what we're currently using now, the artemisinin combination therapies. That's in our country here in Kenya, that's Coartem as the first line, and Coartem is artemether lumefantrine, and the second line is dihydroartemisinin piperacin, and that cuts across most of Africa. In about 2008, in Southeast Asia, artemisinin resistance emerged. And these are the countries in Thailand, Cambodia, and Myanmar. And historically, that's been the hotspot for emergence of antimalarial resistance, and then it spreads to the rest of the globe. So in 2008, when we had this emergence of artemisinin resistance, it obviously caused global concern, and hence the news article I showed you. And then in about 2015 was the first detection of artemisinin resistance mutations in East Africa, and the country that reported this first was Rwanda. So what's the problem? Yes, the drugs are not working. What's the challenge there? The challenge is every time you change antimalarial treatment, there's a cost. So each of these drugs, from the days of chloroquine, which was the cheapest, into sulfadoxine pyrimethamine, into artemethyl lumefantrine, into our current second line, the dihydroartemisinin piperacin, we've gone from 18% increase in cost to about a 400% increase in cost. So that's the challenge with antimalarial drug resistance. The other challenge with it is you have the population that can end up with severe malaria and death. So you'll increase morbidity, you increase mortality, your cost of hospital admissions go up, you have a larger asymptomatic burden who are roaming around in the community and are not being detected, but they're a source of malaria transmission. Obviously, you have hours away from work from people being sick, and as I mentioned, the cost of drug development, but the cost of the new drugs, and your livelihoods are impacted. And in reverse, then your economy goes down and productivity also goes down. And so the main lesson from the early drug chloroquine was when we had resistance, we had a 2 to 3-fold increase in malaria deaths and admissions. So antimalarial drug resistance is a major public health threat. And at the moment, we're at the cusp of another potential disaster if we're not thinking about how we're going to manage this emergence of our current first line and second line, which is the artemisinin resistance mutations. So this is the nearly current landscape of the situation in East Africa in terms of the artemisinin resistance mutations. If you remember, in COVID time, we had variants of concern. So we do have mutations in the gene called KELCH13 in the parasite. And there are some WHO-validated mutations for antimalarial drug resistance, and they're shown in this image over here. And you can see where the arrows are. These are the countries that have detected at least 1 or 2 of these WHO-validated K13 mutations. The current center is in the Horn of Africa and East Africa. There are a few emergence now in Zambia and Namibia, but at a really low frequency. So this is the focal point. And so how can we investigate antimalarial drug resistance? There are about 3 ways. There's molecular surveillance, which I'll tell you about. There's what you can do in the laboratory by taking parasites, adapting them to in vitro culture, and then testing drugs against them and seeing their efficacy in a culture flask. The third way is also to go into the community and actually do a drug clinical trial of your first line and second line. And that's the recommendation of WHO. Every 2 years, you must do these therapeutic efficacy studies to ensure that your first line and second line are still efficacious. The third box, which is the therapeutic efficacy study, is quite costly. You need to go into health facilities and follow up patients from the day you treat them, follow them over the course of treatment, which is 3 days, weekly up until day 28, and then day 42. And during that period, you're collecting blood samples. So what's the scenario we want to see is what you see on, I believe, your left is parasite clearance. So when you have parasitemia on the day you're diagnosed with malaria, by day 3, after taking your treatment, your parasitemia should go down to 0. In some scenarios, we're seeing delayed parasite clearance. So you'll see in the image on your right, some of the lines go down to 0, so you're clearing your parasite, but some of these don't hit 0, which means you have this delayed parasite clearance. It's not as per expectation where we're clearing infections in 3 days. And so this is called delayed parasite clearance, and the WHO say if you have more than 10% of the individuals with parasites on day 3, you should be alarmed and you have a problem. So how are we thinking about mitigating against this problem or controlling is by using genomics as an early warning system to identify these mutations early, and then you can put in some targeted interventions. And so we've developed in our lab in Kilifi anti-malarial drug resistance panel, where we're looking at anti-missing resistance mutations, but for other drugs as well. And we're using the Illumina and the ONT platforms, but essentially what we're able to do is generally in a malaria infection, you won't only be infected by one genetically distinct clone, you'll be infected from anywhere from 1 even up to 12 in high malaria transmission zone, or even 15. And so with next generation sequencing, for each individual, you can identify all of those genetically distinct parasites that are depicted by the colored lines, where we're able to detect all 5 parasites in this scenario. What we're also able to do is not collect a blood sample, but collect a blood sample on filter paper, which makes transportation of samples very simple in our setting, so you do not need cold chain. So there's 2 ways you can use this assay. I talked about the therapeutic efficacy studies. I'm showing you on the left the old method that well, it's not old, it's the standard method we're using, but it's very subjective. You have to use the old capillary sequencer and look at these fragment images by eye and determine whether you have a fragment size or not. What we're trying and hoping to push for is saying use the next generation sequencing platform where you can quantify those infections when you start treatment based on the colored lines, green, pink, and red, and follow them over the course of treatment, and then look at the parasites post-treatment where you have 1008 hours, which is about day 35 or day 28, and you have a different set of colors. So we're able to then quantify how many parasites did you have at treatment and how many parasites do you have following treatment and you've cleared treatment. And that's a more effective way of assessing whether your drugs are working or not in comparison to what I've shown you on the right. So there's some methodology improvements there that we're hoping to work towards, which would be simpler and more quantitative. The other way we're using the tool is on cross-sectional school surveys. So we go across Kenya in the primary schools and we sample individuals, and this is largely the asymptomatic population. So these are children, they're in school. We do an RDT, they're positive, which means they are malaria parasites. We then treat them, but we've got the blood sample on a filter paper that we can use to then screen for these drug resistance mutations using our drug resistance panel. And so what we've been able to show, we did it in 2019 across Western Kenya, the 8 Western Kenya counties. We didn't observe any mutations. We did it again in 2022, but at scale. So as you can see on this map, we've included Turkana, West Pokot. We've gone to Kilifi and Kwale and Tana River. And so if you look at the map, the pie charts, where you have a bit of color, that's not gray, because gray is wild type, but if you have color, it means you have these artemisinin resistance mutations. So in 2022, even though the frequency was low, in most of these counties, about less than 2%, you can see from the dots that we have a distribution of these WHO-validated mutations across the border with Tanzania. The school survey was also done in Kilifi County in about 2021 and 2023, and I'm just showing that there's a difference. Along the western corridor of the country, we have mutations, but in the east, we have 1 or 2 mutations, and these were sampled in 2021 and 2023. So there is some geographical differences where we're seeing higher levels of these mutations in the western part of the country and much less in the east. But when we looked at this in symptomatic, so the school survey is asymptomatic, when we took a set of samples from symptomatic patients showing up at primary healthcare facilities where we do surveillance, we didn't observe any K13 mutations. So again, just speaking to the fact that we have some geographical differences. So the assays, the school surveys were then repeated again in 2023, and so on the top panel, I show you the 2022 results, and I split each one by the WHO-validated mutation. So you can see 4 panels. Each of those are a variant or a mutation that's WHO-validated, and then the lower panel is 2003. So the idea behind this is to look at the spatial distribution of these mutations, and you can observe the third column where you have the 675 variant. It's very low in 2022, but by 2023, you're seeing it across all those 8 counties with a slight increase in prevalence to about 5%. The others are a bit random, where you see them like 561, the first panel. You don't see it in 2022, but you see it in 2023. And for the other two, it's a bit random. So you're seeing low frequency and then you don't see any. But to note here, this is asymptomatic infection. So it's what you see in the community if you go and sample. But the point is, mutations are present in the community. And because these are the populations that transmit malaria, mosquito will come and bite these individuals, it is a cause for concern. So we shared this data immediately with the National Malaria Control Program to provide for them that early warning system and let them know that we have these mutations in our country. So what that allows them to do is then think about doing these therapeutic efficacy studies to determine whether the mutations actually lead to clinical failure. So where I showed you partial resistance and some people are not clearing their parasites. So that's a pending piece of work. So in the meantime, what we're doing is upgrading that panel. It worked really well in terms of doing a single PCR, but now we want to increase our high throughputness of it and do a multiplex. So instead of doing each of the genes or resistance markers as a single PCR, we want to do it as a multiplex and then push it down the pipeline to do the NGS sampling. In addition, because we would like to see if you detect any mutations, we noticed that they're on the border with Uganda. Are any of these mutations coming from across the border, or is it just an independent emergence or within our country? And so we'll do whole genome sequencing as well to look at the whole genome and look for markers of relatedness between the parasites. The other way we're looking at this is using a more inexpensive tool, which is the Oxford Nanopore technology, which gives you slightly less reads, but allows you to do it more cheaply and at a shorter time frame. So the Illumina takes you about 3 days to do the run. With the ONT, you can do it in 24 hours. So we're thinking around utilizing this and potentially moving it to the National Public Health space, where they can routinely then do the molecular surveillance and turn around data. So in the laboratory, we've been optimizing the drug resistance panel on the ONT and thinking through also some quality control steps to ensure that when we detect any mutations, we can talk about the accuracy of these mutations. So what does that look like? In the experiment we did, we took some laboratory isolate controls, mixed them up in different ratios to look for the different genetic markers. So 3 genetically distinct laboratory isolates that we could pick up on ONT. And essentially what we're showing, if you look at the image on your left, is when you have an equal ratio of 33, 33 of each of the markers, you're able to detect them. But if you look at the ONT, it's always lower, which is the purple bar. As you decrease the ratio and go to about 5 and 1%, ONT is struggling a bit when you look at, again, the final section of the first panel. And if you go any lower than 5%, at 1%, ONT does not detect anything. So we're looking at the limit of detection of these assays to look at these low frequency variants that might be emerging as we're thinking about these artemisinin resistance mutations. The other thing we also observed with Oxford Nanopore technology was when we look at diversity of the infection, we tended to pick up some spurious variants. So in the image on your right, if you look at the purple bar at the top, so you have the green and orange, but you have a purpley pink bar, that's a variant we're not expecting to see. So there's some error in this assay. So as we're calling our variants using these technologies, we have to be really careful about the accuracy of the assay and ensuring we have quality control steps. So the idea is, as you're running your malaria molecular surveillance, you ensure you have these quality controls so you can determine your limit of detection. And so, yep, what's the purpose of all of this? It's great to do develop tools and to use malaria molecular surveillance or genomic surveillance to understand what's going on in the background. So I've already demonstrated that. Why we're doing this is because we would like to push the malaria molecular surveillance to scale, which is do it across the country. We would like the National Malaria Control Program and the Ministry of Health to take this up as a routine surveillance so they can set up their own early warning system. You need to be able to count your problem, to put in control measures, and to target what you're doing. And so we've been working with the National Malaria Control Program through technical working group, just bridging the knowledge gap around malaria molecular surveillance, what's the utility of it, through generating a database of all the historical data we have from the chloroquine days into the sulfadoxypyrimethamine and to current, and we've generated a database for them, showing them where the gaps are. We've done trainings on genomics, on PCR, and also together developed this strategy document for malaria molecular surveillance. And the idea about this is to integrate malaria molecular surveillance as a routine activity within the Ministry of Health so that we can roll it out across the country. This is a map of Kenya showing you the regions where you have a lot of malaria. So where you have red and orange on the west are the high malaria transmission zones. Where you have yellow in the east is moderate to high. And then the green bits, you have a low risk. The map has been updated because Turkana has moved it into a high malaria transmission zone. And so what we'll be doing is setting up Sentinel Health Facility sites that will routinely collect these dry blood spot samples, send them to a laboratory network so you can maintain your early warning signal and be ahead of this anti-malarial drug resistance problem and be able to then mitigate and control for anti-malarial drug resistance. And so there are lots of people to thank for this, the funders, the collaborators who helped us develop the tools and provided access to samples and helped us to engage the different communities we work with and stakeholders. And just finally for the institution where I get the work done. And thank you all for listening.
Thank you very much, Professor, for that wonderful presentation. So my name is Dr. Clement. I'll be co-chairing this session with Dr. Mercy. So in the interest of time, I'll only take one question, and then I'll invite the next speaker online.
Just to clarify, why was there a discrepancy between 2002 and 2023?
2022 and
2022 and 2023.
Discrepancy in
the data that you showed. Why was it less in 2022? The molecules that you tested, why was it more in less in 2022 and more in 2023?
Okay, thank you. So what you're talking about is the school survey that we did to look for anti-malarial drug resistance markers. In particular, we were looking at artemisinin resistance. And the reason it's less in 2022 is because it was emerging. When we did the sampling in 2019, there was no mutations. We sampled again in 2022 and we picked up a very low frequency. And by 2023, when we repeat the assay, the number of mutations are increasing. So the issue is one has to do another survey to determine whether the mutation frequencies are still going higher. And if they are, then there's a cause for concern because we are then going to see a high number of resistance mutations that we would need to put in malaria controls measures to mitigate against. Yeah. So it's an emerging trend. Yeah, thanks.
Okay, thank you. So let's applaud Professor. So I want to take this opportunity to introduce our next speaker online, Dr. Nina Billows. Dr. Nina Billows works with the London School of Hygiene and Tropical Medicine. So the presentation on.
Hello, can you all hear me and see my presentation okay?
Yes, go ahead. Thank you. So
Brilliant, thank you so much. Well, thank you very much for having me. My name's Dr. Nina Billows and I'm a research fellow at the London School of Hygiene and Tropical Medicine. And today I'm going to be talking about integrating artificial intelligence for drug resistance prediction in tuberculosis and geospatial classification of malaria. So just to give you a brief overview to this presentation, I'll discuss the background of AI for infectious disease genomics and discuss two applications. So predicting drug-resistant phenotypes from Mycobacterium tuberculosis whole genome sequences, and also the geoclassification of imported malaria cases from Plasmodium falciparum whole genome sequences. And then I'll touch on the future of AI for infectious disease genomics. So just to introduce AI, for those who aren't familiar, artificial intelligence involves systems that mimic human cognitive functions like learning and decision-making. And as a branch of this, we have a subset of AI known as machine learning that learns patterns from data without explicit programming. So this is particularly relevant in the context of infectious disease genomics because it handles complexity and uncertainty in biological data very well. And it becomes very useful for extracting patterns from large-scale, high-dimensional data sets. So there are lots of different models on the market, but there are 7 types of machine learning model, and they can include random forest, logistic regression, neural networks, and also convolutional neural networks as well, which are very useful for image-based classification tasks. So why has AI become so great for infectious disease genomics? So genomic data sets are very large scale and high dimensional. So for example, with malaria, we currently have over 20,000 Plasmodium falciparum whole genome sequences available. So this becomes very challenging to analyze. So with traditional methods, such as statistical approaches, often they struggle with nonlinear relationships, complex interactions between genetic mutations or variants, and AI can excel at extracting these hidden patterns from these very large-scale data sets. So AI has become particularly useful for predictive modeling and classification, particularly using supervised learning techniques. So genotypes are often associated with phenotypes of interest. So for example, we might be interested in drug resistance, for example, in tuberculosis or malaria, or also geographic origin for malaria as well. And so several challenges have emerged about the application of AI to infectious disease genomics, such as how do we connect this genomic variation to observable traits? That is AI actually any good at predicting these phenotypes? And we have to also address several limitations associated with predicting genomic data. So before we heard about genome coverage, for example, that means that we can be missing genome mutations within our data sets. So how can we deal with that in an AI context? We also have confounding and bias. So when we're curating these very large-scale data sets, often the data has been curated and collected from studies that may be focused on a single geographic region, for example. So how do we deal with this in AI? And also interpretability. So we're not just interested in having a really high-performing, accurate model, but we also want to understand the genomic mutations that drive the phenotypes for genome surveillance. So I'm going to touch upon a project that looked at predicting drug-resistant phenotypes from Mycobacterium tuberculosis whole genome sequences, and specifically the mutations in those sequences. So just to introduce tuberculosis, tuberculosis is caused by members of the Mycobacterium tuberculosis complex. And this is divided into several major lineages, and they're constantly discovering new lineages over time. And these particular lineages have certain phylogeographical associations, and they can vary in virulence and also their transmission. TB is spread via airborne droplets and can cause latent or active pulmonary and extrapulmonary disease. And of course, with many bacterial infections, drug resistance has become a key issue. And this resistance primarily arises through the development of chromosomal mutations. There are key types or classifications of drug resistance in TB. So these include multi-drug resistant TB, which is defined as resistance to the 2 first-line drugs, so isoniazid and rifampicin. But unfortunately, we also see more severe resistance developing, and these are often known as extremely drug-resistant TB cases that tend to be resistant to additional drugs, such as the second-line treatment. So traditionally, drug-resistant TB has been diagnosed using drug susceptibility test methods, but these are incredibly slow and require resources. So Mycobacterium tuberculosis is an incredibly slow-growing pathogen, and this makes it quite challenging to actually perform these tests, as it takes months really for somebody to get diagnosed with drug-resistant TB. So newer molecular-based technologies have been brought to market. You may have heard of GeneXpert, for example. However, with these tools, while some of them are very good and very sensitive, we're constantly chasing the genome mutation to detect. So it's been put forward that whole genome sequencing is going to be incredibly valuable. We can use the AI to predict the drug-resistant phenotype, but at the same time, be undergoing that genome surveillance to identify the mutations that are causing resistance, particularly to some of those newer drugs on the market. So within the literature, there were many machine learning models that had been published, and they'd shown moderate to high performance for predicting drug-resistant phenotypes from Mycobacterium tuberculosis genome variants. However, there were sort of several limitations that kept cropping up. So the first was confounding due to population structure. So I mentioned earlier that the Mycobacterium tuberculosis complex has a clonal population structure. It's comprised of many lineages. And you have an uneven distribution sometimes of that drug susceptibility test data available for those lineages. And what we were finding was that lineage-specific mutations, so mutations only observed in a single lineage, were often being prioritized or assigned high importance in the model. And this isn't always the best thing to happen because they don't necessarily cause the resistance. We also have lots of single label learning models. So this is predicting resistance to a single drug at one time. Now, this again isn't that useful because for TB, we use several drugs to actually treat it. So we need to be predicting resistance across a whole panel. However, I'm not going to touch on this limitation today. We also lack interpretable decisions. So as I mentioned, we're really interested in leveraging machine learning prediction to draw actual useful biological insights, like which mutation is actually causing the resistance. So this particular research project aimed to overcome some of these limitations through primarily reducing lineage dependency or confounding due to population structure in a random forest model using what we call a feature weighting approach, which I'll talk about later on. And just for those who aren't familiar, I understand AI is quite a complex topic. Random forests are what we're going to be working with today. So these are ensemble methods. They're comprised of several kind of decision trees, and they've been shown to be very good at genotype, phenotype predictions. And I really like them because they're inherently interpretable. So we can look at which mutations it's assigning high importance to within the model that seems to be driving the prediction. So the first step was to take about 18,000 samples of Mycobacterium tuberculosis and about 1 million variants. Now, this data set has really massively grown now. So we have over 170,000 genome sequences, 50,000 of which have drug susceptibility test data. So we can see how AI is going to become increasingly important in this context. So what we did was we constructed this tree and we saw the very typical clonal population structure comprised of several human and zooxanthellae lineages as well as sublineages as well. And what you can sort of see is that the drug-resistant phenotypes are distributed unevenly across lineages, and some lineages might be considered at higher risk of developing resistance. The drug susceptibility test data is also incomplete. So when you diagnose TB, typically if a sample is susceptible to a certain drug, you then don't go on to test resistance to, say, the second-line drugs. So that can make it sort of quite complex in terms of having a bit of a gappy, missing data set to work with the machine learning process. So as I mentioned, the original aim of this project was to reduce confounding from population structure or lineage dependency when essentially trying to predict drug-resistant phenotypes from the genomic data. And to do this, we use something called a feature-weighted random forest model, where essentially we construct a tree and we derive some weights to put into our model using an algorithm known as Fitcher's parsimony. And essentially this looks at how many times mutations kind of crop up amongst a tree. And if they only, say, emerge once at the base of the lineage, then we kind of downweight them in our machine learning model. We compared this model to an unweighted model and also one that had undergone what we call a traditional feature selection approach, where we essentially just removed all of the lineage-specific mutations from the candidate set. So the actual pipeline kind of looks like this, but essentially we have our global data set and we split it into training and testing data. We then perform some phylogenetic analysis and exploratory data analysis. We process our data, so we essentially choose relevant features. So these will be features in candidate genes or drug resistance genes. We impute sort of missing genomic data. We filter out any missing drug susceptibility test data. And then we essentially go on to make our bespoke machine learning model where we've reduced lineage dependency. We then train and test this algorithm. So when we looked at the performance of this, we found that the performance was generally higher for the first-line drugs than the second-line drugs. And this is something we were expecting because, as mentioned, due to our missing data problem, we generally have a lot more data available for our first-line drugs than our second-line drugs. In terms of reducing that lineage dependency through either traditional feature selection or our new feature weighting method, we found comparable or higher sensitivity when predicting resistance to across all drugs. Now, this was actually quite unexpected because it had been previously hypothesized in the literature that actually reducing this lineage dependency could harm the predictive performance. So it's quite nice to see just because you're removing a confounder doesn't mean that you're going to harm the performance of your model. There's also an expected trade-off between sensitivity and specificity. So I'm incredibly passionate about the interpretability of machine learning models. So the next step was to see, actually, have we improved the interpretability of our models? So what we did was we established some thresholds for the most important features in the model or most important genomic mutations that might be causing us to predict a drug-resistant outcome in the model. And what we observed was an increased importance assigned to known drug resistance mutations using our feature weighting approach. So you can see that by the increased importance in the blue bars. The exception here is for the drug pyrazinamide. Now, pyrazinamide is quite interesting because it has known sort of lineage associations with drug resistance. So this was somewhat expected. We also have a similar observation of improved interpretability for our second line drugs as well. We also observe an increased dependence on what we call co-occurring mutations. So these are mutations that cause resistance to a different drug. So, for example, if we're predicting resistance to rifampicin, we often see, say, mutations in CAT-G that's associated with resistance to isoniazid being assigned high importance. And that's because there's a lot of correlation between these genetic mutations. So that is something that's quite important to consider in the future. So from this project, we found that confounding from population structure remains a key problem, not just for drug-resistant TB prediction, but in general genotype to phenotype predictions. And this has been especially exemplified in the prediction of drug-resistant phenotypes from genome variants of the Mycobacterium tuberculosis complex. So we found that reducing lineage dependency can help boost both the interpretability of the machine learning prediction, but also maintain or improve predictive performance. Future research should also address other confounders, such as co-occurring resistance. And just to touch upon again, it's really important that we're able to draw kind of useful biological conclusions. We want to be looking at some of the drug resistance mutations, not just the predictive performance of the model. So I would also just like to touch on another project we are actively working on at the moment. So in the UK, we have approximately 2,000 imported malaria cases each year. And occasionally we have an individual who's travelled to a non-endemic country and unfortunately come back with malaria. And then there's this kind of challenge of trying to figure out where that malaria must have originally come from. So this is where AI becomes quite useful. So what we've done is we've taken now over 20,000 samples of Plasmodium falciparum whole genome sequences. Again, we've split our data, so we're going to use 80% to train our model, 20% to use to test it. And then we've selected some geographical markers that we think are going to be really helpful for predicting this geographic origin. We've then developed this new model. It's a type of convolutional neural network model traditionally used for images, but we have used them on genomic data in the past, and they've been quite helpful and useful. We've then optimized our model. We're assessing its predictive performance. And what we're hoping to very soon is actually implement this in our genome surveillance tool, Malaria Profiler, which is also very good at identifying drug resistance mutations, amongst other things as well. So I can't touch on any of the clinical data that we've been working on in the UK, but I can show you how this model performs on public data obtained across the world. And you can see here it actually performs pretty well. And the only time we really see misclassification is in a kind of border region. So for example, you can see here, Central Africa might get accidentally mispredicted as East Africa or West Africa. And that probably is the case in the data set anyway. We probably do have some importation amongst those regions as well. So just to conclude and talk about the future of AI. So AI enables large-scale genomic analysis. It can process large-scale data sets to predict things like drug resistance, lineage, or geographic origin. Custom models are essential. It's not a kind of one-size-fits-all approach. I'm a big fan of making things bespoke and as good and accurate as possible. and they should always be trained on kind of relevant pathogen populations and data types. Interpretability is also critical. It's not just about having a really accurate model. The predictions must also be explainable to guide clinical or public health decisions. We also need to consider how to use AI very carefully. It should complement, not replace expert judgment or diagnostic, but it can be useful. And I'm particularly excited for the future directions of AI in infectious disease genomics. So the real-time genomic surveillance, and the production of interpretable outputs and integration with epidemiology to really transform infectious disease monitoring and outbreak response. And just to say a big thank you for all of the individuals who've been involved in this work and all of the samples collected by everybody in these really large scale data sets. Yeah, it's been fantastic. And thank you so much for having me. Yeah, I'd be open to any questions. Thank you.
So thank you very much for that wonderful presentation. So I have one question. In your AI, especially the one that predicts TB, did you include all the reported TB lineages, particularly those that have been associated with resistance to newer drugs like bedaquiline and linezolid?
Yeah, I think that's a really important question. So this particular model was from a slightly older publication, but we continuously update our data sets and retrain our models as well to make them as clinically relevant. With drugs like bedaquiline, linezolid, sometimes it can be quite challenging to actually use AI because we don't necessarily have enough kind of statistical power there. However, it is something that we are kind of moving towards to make our models as relevant as possible, especially for some of these newer drugs. Thank you.
Okay, thank you. So one more question from the...
Thank you so much for that nice presentation. I've realized that from your presentation, you mentioned that you used CNN for your neural talk. So did you do any comparison with other types of neural networks for you to decide on the CNNs?
Yeah, exactly. So I actually do really like using some of the shallow approaches as well. So we have things like random forest, logistic regression, and it's a kind of special kind of type of CNN as well. So it's actually a multitask. And that's the reason why we've kind of gone for that particular model. But yeah, we have played around quite a lot with different parameters, different types of neural network. And it's something that we're kind of actively developing at the moment. So it's kind of changing over time. But yeah, fantastic question. Thank you.
Okay, let's applaud Dr. Nina Billows. So at this point, allow me to introduce our next speaker, who's joining us online again, Dr. Oluwafemi Oluwole from University of Cape Town, South Africa. Doc, can you hear us? So a bit of a technical hit. So allow me to introduce our next speaker, Dr. Farad Zamani from Aarhus University, Denmark.
waiting for the slides, I will introduce myself. I'm Farhad Zamani. I'm representing Aarhus University in Department of Molecular Medicine, and also affiliated with Aarhus University Hospital. And in today's session, I will explain and touch upon generative AI, the use of generative AI in biomedical research. But before that, I will start with a little ice breaking because it's been a long time and it's already quite late in the day. And I will start by showing you this picture. Can you see this picture? Can you tell what this picture is? And maybe I will give you some hints more. What about this? And the next picture. Yes. It is a picture of cats, and you can also guess from the first picture that it looks like a fur of a cat. Then this morning also, because I'm bored, I asked ChatGPT to generate a cat image, and this is what they gave us, what it gave me. And then because I don't like the color of the cat, I asked them to generate the same cat image, but with white color. And ChatGPT gave me this picture. It is quite cool. But the question is, how can a machine learn the properties of cat image? Because you know that cat has a super specific pointy ears, and then a cute paws, and then also the tail. But how can ChatGPT know the properties of cat? To put it simply, in general, they learn, the model is trained by using a lot of pictures of image. For example, this is the picture of the cat, and then it fed into a machine or a model to give us an output image, which is also a cat. And behind this model, it is, in a simple way, it is basically a neural network and some modification of neural network. And there's a specific type of neural network that is called autoencoder. It basically fits the same picture, and then there is a latent space, latent vector to get a set of numerical features that represent a set of features that is important, that best represent the image. But it can only reconstruct the same image of, like the same as the input. However, there is also some modification of this type of network called variational autoencoder. It trained upon a lot of different cat images, and then it tries to learn different properties of the cat, maybe not exactly the properties, but it captures a set of features that is important. And then it can, and then it learns the distribution of the properties as well to generate a different image cat. And that's, we call that type of model as a generative AI model. It learns the underlying distribution of the data, and it can basically be any data. It can be cat, dog, or your biomedical data. So it learns the distribution of the data, that's, which you can see in the left, and then there is some function. It can be neural network or other type of functions that map from the underlying distribution to a data space, which you can see in the right side. And then another question, can we use this type of generative AI modeling in biomedical research? Because you're not only want to you don't only want to generate cat image, right? You want to use it in your biomedical research or maybe your work. And then I will give you some questions. That is, I will give you some examples. That is my project currently that learns the regulatory mechanism of microRNA expression. this microRNA, it regulates, check. Check, yes. So this check. Yes, super. Now we get back. This microRNA, it regulates the expression of mRNA. Yes. So in our cell, in our body, and it targets a different mRNA. Check, yes. It targets a different mRNA, so it will inhibit the expression of mRNA because it is degraded. So in our group, we are working mostly with cancer data, and microRNA and mRNA expression, correlation, and relationship structure is important because there is a dysregulation of microRNA and mRNA in a cancer sample. So it is important, and it is important to study this kind of regulatory interaction. And we can usually sequence microRNA and mRNA in bulk settings. So from the tissue, the sample is the bulk tissue. But in a single cell RNA sequencing setting protocol, microRNA is hard to quantify. So we try to figure out how to use of this generative AI model to help us to do this task. And I built a model, generative AI model called MyDGT. It is a generative AI model to predict microRNA expression only from mRNA expression. So basically, it takes mRNA expression and microRNA expression, and it fed into the model, and then it will make a representation, low-dimensional representation of the data. And then from the representation of the data, which you can see also in the previous slides when I show you a cat picture, the model will map this low-dimensional space into a data space, which is a high dimension. Yes. And to make the model works, I used a paired mRNA and microRNA expression profiles. So it comes from the same sample, from bulk sample and single cell sample, in single cell sequencing. And we use the same tissue or cell to get both mRNA and microRNA expression. And mostly we use public data set, such as TCGA, the Cancer Genome Atlas, GTEx, RNA Atlas, and the SmartSeq 222 and PSCRSeq for, it is a detail, but that data is available online. And we also use a lot of different tissue type and cell type, and try to learn to train the model whether they can differentiate between tissue types and cell type. And then this is the detail, but basically it takes gene expression or mRNA expression and microRNA expression to this whole machinery to get the representation, which represented as Z, to do some inference. So because we don't have microRNA expression, we only use gene expression to predict microRNA expression. And then after we have the trained model, apparently, the model can capture the low-dimensional representation of the dataset. In this case, I'm only using a subset that is a TCGA dataset, and it is unsupervised, so the model doesn't know where the sample comes from, but it can find a latent structure and one dot represent one samples, and it can cluster based on cancer type, which is quite nice. And then for the main task, which is predicting microRNA expression, I choose this microRNA expression mirror 120 to 5p, so in the 5p arms of the microRNA from the liver hepatocellular carcinoma. This microRNA is specific, tissue-specific, that is mostly expressed in tissue types, liver tissue types, which is also shown in the plot that I show you in the right. And it doesn't predict any other cancer types that is not from the liver. That is quite good result. And then this is the overall general overview of the correlation. Previously, it is from 1 microRNA, and then we have around 700 microRNA that we want to predict the expression of it from the mRNA. And this is the correlation both for Spearman and Pearson. And then we also want to predict microRNA expression from the single cell setting. This previous slide, we are predicting on bulk RNA sequencing setting, and this is from the single cell mRNA expression settings. And we also got quite high, I would say it's quite high, mean expression of Spearman and Pearson for this. And this generative AI modeling, and you can also use any type of model, it gives a promise that, not promise, but there's a lot of potential to use this kind of generative AI modeling for your biomedical research if you have a lot of data to train the model on. That's all. Thank you. I want to acknowledge the Skov-Pearson group from Department of Molecular Medicine, Aarhus University and Aarhus University Hospital in Denmark, and all the co-authors and also the funders. And I'll be happy to take questions if there's any time.
Thank you, thank you, thank you. So we can take one question.
Hello. Yeah, great talk. I pretty much enjoyed it. Now, rather naive question, so you will excuse me, but I was just wondering, when you try to correlate mRNA expression and microRNA, I was just wondering, what's the assumption you're making? Are you making the assumption that the expressions occur simultaneously, or they are synchronized? What's actually the underlying assumption that you're making?
So,
if that is relevant to the model.
Yeah, that is a good question, actually. So we fit the data without having to assign which mRNA targets mRNA. So we don't know, we don't fit that information, but the model works under the assumption that there is a correlation structure between microRNA and mRNA, and maybe vice versa, because if the expression of microRNA is increasing, you can tell that maybe it binds to a lot of mRNA, so the expression of the target mRNA is decreasing. So based on that correlation, we built this model, and apparently the model works to predict some microRNA that is well-defined. Maybe they have a well-defined regulatory mechanism in the cell.
Okay, so another question, just a quick one. Is it possible to actually also identify the functionality of the microRNAs? There are some that are functional, or some we still don't even know what they do, because inside the cells they're quite abundant microRNAs, and I was just wondering, are you able to predict any functionality based on your model?
Based on this modeling, I don't think we can predict the functionality, but we can see to which to which microRNA or mRNA that this microRNA, specific microRNA can have some maybe correlation between other mRNA and microRNA. So maybe based on that we can go further to check whether it actually targets that microRNA, that mRNA, that they have a correlation among them.
Okay, thanks. Great. Thank you.
Okay, thank you.
Thank you so much.
Okay, I want to introduce our last speaker for today, Dr. Christian from Swiss TPH. So welcome.
Good evening, everyone. First of all, I would like to thank the organizer for giving me the opportunity to present some of our work in our group on malaria. I think actually Isabella gave actually a nice introduction to malaria molecular surveillance, and I will be following up on that to show some of the work we've been doing also on malaria molecular surveillance in Rwanda. So the project I'm going to talk about is also about therapeutic efficacy study and how you can use actually samples collected from these clinical trials to also use molecular markers of resistance to assess how resistance is spreading in a specific country. Yes, so this is the title. It's about resistance markers, not only K13 associated with artemisinin resistance, but also mutations associated with partner drugs, because you have a combination of artemisinin derivatives and partner drug. And to have resistance to ACTs, you need to have actually resistance to both components. And this work actually was a collaboration between different groups, the Swiss Tropical Institute, but also the PMI, the President Malaria Initiative group at CDC, and also the Rwanda Biomedical Center. Again, Isabella gave a nice overview of malaria and where we are now. We are now actually having sometimes even in some countries an increase in malaria cases, despite the progress that have been made over the last 10 years. So 2 years ago, we had around 200 million cases in 2023, with approximately just over 500,000 of deaths due to malaria. And these were mainly caused by Plasmodium falciparum, that is causing most of death, mainly in children under 5 years of age. And the main actually control tools we have at the moment are bed nets, indoors residual spraying with insecticides, and also improved case management with efficacious drugs, artemisinin-based combination therapies, but also chemoprevention, so using anti-malaria drugs to prevent malaria, because until recently we did not really have vaccines. We have 2 vaccines that have been approved in the last 2 years, but their efficacy is still quite not optimal, so we are still using drugs to prevent malaria. So, but some issue, as also Isabella alluded to previously, we have resistance that is developing not only to artemisinin, but also to partner drugs. We have actually that partial artemisinin resistance that has been confirmed in countries in East Africa. And we have also insecticide resistance that is widely distributed both in West and East Africa. So then looking only at malaria in Rwanda, so there were a peak of cases actually in 2016, where there was an incidence of more than 200 cases per 1,000. And then there was a large campaign to distribute bed nets and also to deploy indoor residual spraying in most of the districts in the country. And then by 2000, actually a lot of progress have been made. And there were around 76 cases per 1,000 inhabitants, and are now currently actually 25 of the 30 districts. They have an incidence of below 100 per 1,000. But the entire population of the country is still at risk of malaria, and there are some groups that are at high risk of malaria because they have low coverage of these different interventions, including IRS or bed nets. And also some districts, actually, they still have a high burden of malaria. And of course, the partial artemisinin resistance that has been reported a few years ago. And for this study, we used samples that were collected from a therapeutic efficacy study. So these studies actually are meant to assess the efficacy of the anti-malaria drugs that are used in the country. And as for Kenya, in Rwanda, the first line treatment is artemether lumefantrine, Coartem, and the second line is also dihydroartemisinin, piperacin, urartezim, or diocotexin. And we collected 256 samples from these patients. So you can see the 3 sites. Samples were collected actually at one site in the western part of the country, close to DRC, at one site in the capital, Kigali, in the center of the country, and at another site in the eastern part of the country that is bordering Tanzania. And those samples were used for 2 different analyses. We looked actually at the markers that are used to assess recrudescent new infection. As you may know, when you assess drug efficacy for anti-malaria drugs, you need actually to treat the patient and follow them up to 1 month or 2 months, depending on the drug. But you can have reinfection by new mosquito during that period. So to make sure actually that you don't underestimate the efficacy of the drug, you need to differentiate this new infection from true treatment failure, and that you will have actually what we call PCR-corrected efficacy, where you remove the new infection and only keep actually the recrudescence. We use those samples also to look at molecular markers associated with drug resistance. So we collaborated, as I mentioned before, with the Rwanda Biomedical Center and the PMI team at CDC. And actually the findings that were first analyzed by the CDC team were published in 2021. And that study confirmed actually that there was delayed parasite clearance in the country associated with PFK13 mutation. So therefore, there was really partial artemisinin resistance in the country. And then for doing this PCR correction, they use these classic methods of capillary electrophoresis using microsatellites and also use Sanger sequencing to look at PFK13 mutation and PFMDR. So then after that, we obtained these samples from CDC and we continued the analysis. And then we used other technique actually to differentiate this recrudescent new infection. We continued with capillary electrophoresis, but we moved to MSP1 and MSP2 instead of microsatellites to see actually if you are using different molecular markers, you can get similar results. But we also used amplicon deep sequencing using Illumina, and for that we used 4 different markers. And we completed actually the analysis of resistance markers looking at PFCRT mutations, PFUBP1, DHFR, and DHPS. And this study was done at Swiss TPH with a master's students and 2 postdocs that were supporting for the bioinformatics analysis. So when we look actually at these molecular markers of to differentiate new infection, we tested different microsatellites to see how they perform in terms of sensitivity. So similarly to the study that was shown before by Isabella, we mixed different lab strains at different ratio, and we're trying actually which microsatellite was good in detecting the strain that was in minority. because people have realized in this polyclonal infection you find in many malaria endemic countries in Africa, you always have more than 2 clones in the same infection. And for a good technique to be able to differentiate recrudescent new infection, you need to be really sensitive in detecting those minority clones. And when we look at that, for example, we found here that the microsatellite PFPK2 was very good in detecting minority clones, as it could detect up to 1, actually when the minority clone was at 1 and the majority clone was at 50 times more than the minority clone, this marker actually could still detect the minority clone that was at 1 ratio. We looked also at reproducibility, so that means we repeat the same assay over time, and we also ask a different technician to do the same assay, and we see how many times actually you get the same results. And here also we had the microsatellites PFPK2 that was performing best, giving actually a good reproducibility rates, even though actually it's quite low compared to, for example, other markers that's like amplicon sequencing or MSP1 and MSP2. And at the end, we also looked at the genetic diversity and allelic frequency. So if you want a good marker actually to differentiate this recrudescence and new infection, it has to be very diverse. So it has to be able to distinguish the different clones of Plasmodium falciparum that are circulating in the population. So that means here when you have this N, that is the number, so that means from the same number of samples, this is the number of different clones you can detect with these different markers. And here you can see, for example, with 3D7, you can detect up to 65 different clones. But when you look at microsatellite, and this is the same samples, you are only detecting around 20, 23 different clones. So that means actually the microsatellite do not have a good specificity in differentiating different clones. And you are also trying to look that no clone has a very high prevalence, because if you have here, for example, some of these microsatellites, they show a prevalence of some clones above 20%. So that means actually it's not good. You will have a lot of clones that are different, but they have the same genetic profile when you use the microsatellites. And this we could see also when we use the markers with amplicon deep sequencing. You also have a high genetic diversity, and they also show that you have a high number of clones compared to the classic techniques like MSP1 and MSP2. And then when you have different markers actually to differentiate these recrudescent new infection, you always use at least 2 or 3 different markers. And then to get the final outcome, you can use different algorithms. So that means if you have 3 markers, 2 are showing it's a new infection, and 1 is showing it's a recrudescence, how do you decide actually at the end if it's a new infection or not? So currently the WHO does recommend that to show that it's a recrudescence, all the 3 markers you are using have to show actually this infection is a recrudescence. But people have been developing different algorithms, some based on Bayesian statistics, to take into account actually that the facts, the markers do not have the same sensitivity, the techniques do not have the same also sensitivity to detect minority clones. And what was interesting here, we found that when you use these amplicon sequencing markers, actually the final outcome does not depend on the algorithm we are using, because all the markers used tend to give the same results, and then they have a more consistent results. And therefore, actually, we think in the future we should more use these amplicon sequencing techniques, because they have markers that are very sensitive, very diverse, and also giving really consistent results compared to other markers. So even though at the moment we've done that study only in one country, in Rwanda, so I think this should be repeated in different countries to make sure actually this is generalizable and you can use all those markers in different countries. So moving forward, we've looked also at the resistance markers. So in the initial study that was done, the PMI team at CDC, they found a high prevalence of PFK13 markers that was associated with delayed parasite clearance. And as we followed up, actually we looked at another marker that is called PFUBP1. And this marker has been also associated with artemisinin resistance in some studies. And what was interesting for us, it was basically in the sites where there were a high prevalence of K13 mutation, the prevalence of this other marker also associated with artemisinin resistance was low. And where there was a low prevalence of K13 mutation, the prevalence of PFUBP1 was high. And that's quite interesting because recent studies have shown that K13 and PFUBP1 are both involved actually in the same pathway and are used actually by the parasites to take the hemoglobin from the red blood cells. And both actually proteins are probably involved in hemoglobin degradation and the production of protein in the parasites. We looked also at the PFCRT mutation. We know, for example, that wild-type PFCRT is associated not with resistance, but slightly decreased susceptibility to lumefantrine, so the partner drug of artemether in Coartem. And we could show also that most of the samples collected had a wild-type haplotype of PFCRT, suggesting also that the susceptibility to lumefantrine may be decreasing in the country, which could at some stage actually lead to treatment failure with Coartem. We looked also at other markers like PFPM2, so this is plasmepsin-2, and this marker is associated with resistance to piperaquine, so this is the second-line treatment. We could find, actually, we have not finished the analysis of these samples, but there were actually a few samples with increased copy number of plasmepsin-2. Here, we wanted to show just the control with the arrows. So these are lab strands with non-copy number of plasmepsin. So we have 3D7 that has one copy. And then this is an isolate from Cambodia that is known actually to have 2 copies of plasmepsin. So here we found actually 3 samples that contain isolates with plasmepsin-2 amplification, and 1 sample with PFMDR copy number with 2 copies of PFMDR. Going on, we followed with the prevalence of PFDHPS and DHFR. So these are markers associated with sulfadoxine pyrimethamine resistance. So SP, even though it's not used for treatment, it's still used a lot for chemoprevention. So that could be for pregnant women, intermittent preventive treatment in pregnant women, but also in young children for perennial malaria chemoprevention or seasonal malaria chemoprevention. And here actually we found a very high prevalence of both PFMDR mutations, but also the combination of both PFDHFR and PFDHPS mutations, especially what we call the septuple mutations. That means we have 4 mutations in DHFR and 3 mutations in DHPS, and that means actually you have the DHFR 164L mutation that is really leading to very high level of resistance to SP. And that prevalence actually at one site close to Tanzania was at 25% level. And this was quite intriguing because Rwanda does not use any more SP, and they don't even use SP for chemoprevention. So IPTP was discontinued more than 10 years ago because of high level of resistance. There is no perennial malaria chemoprevention. There is no seasonal malaria chemo prevention. And even recent studies have shown actually you don't really find SP in shops or in clinics in the country. But when we look actually at also previous data, it seems the level of SP resistance is still increasing in the country despite the fact that the drug is not used. So we had some hypotheses. It could be actually like maybe it's coming from neighboring country. We found actually a very high level of resistance to SP close to Tanzania where IPTP is used, and this is something that should be investigated. But also the use of cotrimoxazole, this is an antibiotic that also could lead to resistance to SP. But again, on unauthorized use of SP, it seems that it's highly unlikely because you can't really find it in the country. So we are continuing that. So we have now started a study to look at the flanking gene of the flanking region of these genes, PFDHFR, DHPS, and try to compare from samples coming from neighboring countries to see actually if those samples with this haplotype that leads to high level of resistance are originally from Rwanda, or they also came from neighboring countries. And these actually, we wanted to show like molecular surveillance at the moment is only used, people will take samples from TES and then try to look at resistance markers as we did in these studies, but we should think beyond that. We should really aim at expanding molecular surveillance on a country level, regional level, and to get actually a more precise image on how resistance is spreading not only on a country level, but on a regional level. And this is like a good example from a study that was done in Uganda, where they looked actually in the whole country, what is the prevalence of the PFK13 mutation, those validated by WHO, and how that will evolve over time. And that can inform actually countries where they can do these therapeutic efficacy studies when they see there is a high prevalence of mutation. And these actually, we can use molecular markers as an early warning tool for clinical treatment failure. And these are actually data that were collected, I think, from Kilifi, showing actually over time resistance with SP. That's when you look at SP resistance in vivo, that means with clinical trials, it happens after the resistance markers, so the mutation in PFDHFR and DHPH had spreads, and also the in vitro, so IC50 had increased. So that means actually when you use these molecular in vitro markers, you can predict when treatment failure will emerge. So just coming to an end, so you have these different use cases for malaria genomics. You can use on a country level or regional levels, so looking at recrudescent new infection, diagnostic resistance with HRP2 3 deletion, but also insecticide resistance and drug resistance. So to finish, I would like to thank our collaborators from the Rwanda Biomedical Center, but also the PMI initiative and CDC team, and also our group, the Swiss Tropical Institute, that was also involved in generating this data. Thanks a lot.
For the wonderful presentation. So Dr. Christian is still here with us, and I would want to encourage anyone with a question to kindly ask him. As you can see, it is actually 8 p.m. Ladies and gents, allow me to close the session at this point. Let's applaud all our speakers for the day. So tomorrow we converge here at exactly 8 a.m. so that we can save on time. Thank you very much.